Let's go ahead and get started. We are halfway through the, I say semester, but really summer right now. We are starting our third section of the class. This is going to be the muscular system. Again, as we talked about before, there are 4 tissue types: epithelial tissues, connective tissues, muscle tissues, and nervous tissue. We've talked about epithelial and connective tissue. So today we will be talking about muscle tissue and muscle anatomy. And then that will give us the basis for all the physiology we're going to be doing with the rest of this section. And that'll include talking a lot of histology, not as much as the epithelial tissues and connective tissues, but more than we saw in the bones. A handful of assignments. First, Monday is our holiday, so we do have a nice 3-day weekend. So I know you may be a little hungover from the exam from yesterday, but you'll have a nice 3-day weekend to get ahead of the material, and I strongly do encourage you to use that time for that. As a small carrot to encourage you to do that, you do have a Labster activity on the muscle histology called muscle tissue. Again, you must complete all of it and get it at least 80% correct for full credit. This section doesn't have quite as much homework because there is a lot of material for us to cover. We have 2 unit reviews, unit review 10 and 11 that are going to be due. We have the Labster, which you're going to be doing. I know you guys had so much fun with the first PhysioX exercises, so we'll be doing those again. I remember there were 5 in the first one, and so you had 5 lab reports. Well, this one has 7. So there's 7 lab reports for that. So again, as you saw, it's not hard, it's just time consuming. So definitely don't leave that as something that you try to do the night before because it'll take a long period of time. And we have a muscle activity handout that you'll be working on, kind of like it's almost like a muscle review, the same way that we had a skeletal review. That will be graded for correctness. So make sure and we'll talk about that as we get closer to that as well. And that's the game plan. We are going to, much like the skeletal system, the muscular system, we will talk physiology in lecture, and then in lab, we'll be doing a tremendous amount of anatomy. Trying to work through that material together quickly, not necessarily to master it, but again, to expose you to it so that you have some familiarity and you're ready to go with that. All right. Questions on any of that?
Yes, Professor. Actually, it was a question more about the exam, this last exam. So during the lab exam, it took forever to do it, loading one screen at a time. Is there any possible way that we could do it all on one thing? It helps out a lot.
As I said when we were going through it, this one at a time is not my preferred method. However, with the first exam, when we had it all on the same page, which is the way I prefer to do it, close to 1/3 of the students had problem loading with that format. The problem is if the Wi-Fi isn't, if the internet connection isn't strong enough, if the amount of RAM available isn't strong enough, then it really drags down and bogs down, and 1/3 of the students weren't able to get it to load. We're having problems. So unfortunately, because of the restrictions of what we're able to do this way, it's unfortunately the format that works best for more people. I do put more time. Typically an exam like this, you get 1 minute per question. And so with 78 questions, really 77 because of the integrity question, you guys did have 95 minutes. So I did try to put almost 20 minutes of additional time to try to compensate for that loading time. But unfortunately, we had so many problems on the first exam, and I don't know why this is happening. We've been online for 1.5 years now, almost 2 years, and I haven't had this kind of problems before, only once before at the very beginning when we were first doing this. But for whatever reason this summer, I don't know why, that there was a tremendous amount of difficulty with that. Like I said, close to 1/3 of the students had some problems. So unfortunately, that is going to be the format for the exams, just because it's the one that, while not ideal, it's the one that at least works so people can take the exam. But yeah, no, I don't care for that format as well, but unfortunately that's the way that seems to work best. And again, honestly, that's the way it is in the classroom. In the classroom, when you're sitting down in the classroom, we would do an exam like that as a PowerPoint presentation. You'd be presented with the questions one at a time and you'd go through them. And in the classroom, you can't go back, at least on this format. You can go back to double check something if you weren't certain about it. So it's ironically the one most similar to what would be in the classroom, but for the online, it's not my preferred, but it's what I feel is necessary for the compatibility of all students. All right, any other questions? All righty, excellent. Let's dive into lecture then. Like I said, our goal is to indeed talk about the muscular system and all of the muscles associated with that. As we talked about, we have 206 named bones, and you had to learn them all. Anybody know how many named muscles we have?
Over 500?
Yeah, over 600, actually, absolutely. Definitely over 500 and even over 600. But don't worry, you're not going to have to learn all 600 of that. You only have to learn about 475. So it's not so bad at all. No, actually, there won't be anywhere near that much, but which I'm sure you know, because I'm sure you've already looked at the handouts. But like we also talked about, before we can get to the muscle organs, we need to talk about the muscle tissue. So let's do that. Like epithelial tissues and connective tissues, they have functions of the body and they had characteristics that they all shared. For instance, epithelial tissues form the barriers of our body. They were superficial, they were cell dense, they were avascular. So we talked about their functions and we talked about their basic structure and anatomy as well. So let's do the same thing for muscle tissue. What are some of the things that muscle tissue does for us?
Body movement.
Excellent. And we've been saying that a lot. We've been talking about from the very beginning that muscle tissue allows for movement. But let's be more precise than that. What do we really mean when we say movement?
It moves our bones.
Okay, so it moves our body through space, or bones. I like that. That's precise too. But what happens if I put my arms out and I hold my breath? Is there still movement going on within my body?
No, but you're stabilized.
True. Okay, so that is another function of the muscle. But is there really no movement going on in my body?
There is.
Like what?
Like oxygen and I mean not all like all the other stuff, the nutrients and
True, those are being moved through the body. How? How's oxygen and nutrients being moved through the body? You're absolutely correct. Anyone?
the blood.
The muscle.
Yeah, there you go. The heart is pumping the blood, right? Even though I'm sitting here with my arms out holding my breath, that my stomach is still working on the cheeseburger that I had for breakfast, right? So when we talk about movement, yes, there's movement of our body through space, our bones moving them through space, but we also have substances moved through the body. And how does that blood move through our body? How is it that
Homeostatically?
Yeah, exactly. Well, what happens here is that you've got the right idea. What makes the world go round?
Cheeseburgers.
Pressure.
Pressure. Pressure makes the world go round, absolutely. And the way we affect pressure is by changing the volume, right? Our heart is a pump. When it changes volume, it changes pressure, and that pressure moves the blood through our body. When our stomach changes its volume, it changes its pressure. So we move through substances through our body by changing the volume of our hollow organs, and that changes the pressure. So we're able to propel substances through our body by changing the volume of our organs. So it isn't just about waving our body through space. Absolutely, that is a movement, but there is more movement than that. There's also the movement of substances through our body. So we have those two different types of movement. Excellent. Now, someone also pointed out, it also stabilizes the body, holding it in place, not just when I'm holding my breath and holding my hands out, but also allowing me to stand upright. there are a tremendous number of minute little contractions and changes in tension that take place in my body when I'm stabilizing and holding my body in space. And we don't appreciate how complicated that process actually is until it's disrupted by something. Maybe instead of a cheeseburger, I had 4 Jägermeisters for breakfast, right? If I had 4 Jägermeisters for breakfast, would I have as easy of a time being able to control those minute little contractions of my body that keep me stable and upright?
Negative.
No. So that body posture is another important function of our muscle tissue, right? And kind of what goes along with that is also not just posture, but stabilizing and supporting our joints, right? When I'm not using my right arm, my humerus doesn't slowly fall out of the glenoid cavity of my scapula. I mean, heck, we've seen that glenoid cavity of the scapula. There's not a lot of bony structure there that holds that head of the humerus in place. It is stabilized by muscles and ligaments and tendons that are holding it. So stabilizing and supporting our joints, holding them in place. What else does our muscles do for us, or muscle tissue do for us?
Temperature regulation.
Temperature. Excellent. Remember way back when we were talking about the skin, we talked about the important role that muscle plays in regulating our body temperature. And the reason for this, I don't remember if we talked about this at the time, but it's important to talk about it now, is that muscle is a motor. Or let's say it this way, contains motors. Did we talk about motors? Do we know what motors do? Well, even if we did, let's remind ourselves. What is it that a motor does?
Combustion.
Okay, what do you mean by that?
Takes
You're right.
energy of a fuel source and converts it into movement or power.
Perfect, absolutely. A motor's job is to basically convert a chemical energy into a mechanical energy. Right? Just like what you have in your car. Which is why you get to the supermarket. You're very, very thankful for your car for getting you there. So you lay down with your head on the hood and you give your car a big hug when you get to the supermarket. Is that what you do? Do you hug your car and lay your face on the hood of the car when you get there?
No.
No, for converting that chemical energy into mechanical energy. Why not?
Hot.
Yeah,
That's its
exactly.
job.
Because while a motor converts chemical energy to mechanical energy, most motors are not 100% efficient. And a lot, especially with our muscles, a lot of that energy is lost as heat. In fact, in some of our muscle contractions, as much as 75% of the energy is lost in the form of heat. That is incredibly inefficient, right? If your car was only able to convert 25% of the energy of your gasoline into propulsion and lost 75% of it as heat, would that be the kind of car you would want to own? No. But here it's okay because, as we've talked about, that energy lost from the heat from the contraction of the muscles plays a huge role in helping us to regulate our body temperature, which is why we shiver when we get cold, right? Not because we need to move our body through space, but moving our body through space releases energy, releases heat. Excellent. I can think of at least 2 more good functions.
Storage of nutrients.
Yeah, absolutely. How so?
Glycogen.
True, it does indeed store glycogen. But is it storing the glycogen for us to use?
For the body.
Yeah, not for the body so much. That glycogen is stored more as a fuel for producing that energy that it needs to move. But again, summer is upon us. So I'm sure like you're trying to get into bikini shape, just like I'm trying to get into bikini shape. And to get in bikini shape, I am on that famous popcorn diet, where I have 1 piece of popcorn for breakfast, 1 piece of popcorn for lunch, and a very reasonable 3 pieces of popcorn for dinner. Now, if I do that, am I going to lose weight as a result of that popcorn diet?
Yes.
Yes. But is it because the fat is just melting off of me?
No.
No. The reason is because I'm starving myself, right? However, your body does something interesting. When you're being starved, your body gets very, very defensive. And one of the defensive things it does is actually to hold on to the fat. Fat is a valuable resource. So it doesn't like to break it down if it doesn't need to. So when you go into starvation mode, your body actually starts to hold on to the fat. And if it's holding on to the fat, then what gets broken down that causes you to lose the weight?
Muscle.
Your muscles. Your muscles, yes, are absolutely chock full with calcium. In fact, outside of the bones, as Samuel pointed out, muscle tissue has the highest concentration of calcium in the body outside of our bones. But in particular, the primary nutrients that are stored in muscles are proteins. Massive, massive amounts of proteins. And so what happens is when you go into starvation mode, we start breaking down those proteins and releasing them into the blood so the body can get the amino acids that it needs to survive. So basically what our muscle acts is as a nutrient reserve. When you're starving yourself, you are much more likely to break down muscle tissue than you are to break down fat to get the resources necessary for your body to function. And the last one I'll give you, protection. This kind of goes along with the stabilization of the body. Obviously, when we think of protection, we think of things like bones, like the skull, like the ribs that provide that protection. But think of your abdominal pelvic cavity. How many bones are protecting all the muscles of your abdominal pelvic cavity?
0.
Yeah, 0, absolutely. I mean, maybe you could argue the very bottom of the ribs kind of hang down and protect some of them a little bit because the diaphragm is curved, but not a whole heck of a lot. And how many organs do you have in your abdominal pelvic cavity? How about a lot?
A lot.
Yeah, you have a lot in there, absolutely. And so they need protection. And the way they're actually protected is a structure we call the abdominal girdle. We have all these layers of muscles at different orientations, providing that girdle, providing that protection, right? That's how Harry Houdini died. Harry Houdini, the escape artist, the magician, he would put on these huge shows where he would do all these escape acts, and setting up for these escape acts took a tremendous amount of time. And so in between his escape bits, he would have other performers who would come out and perform little bits. But one of the other things he would do is he would come on stage, and to show his strength and veracity, one of the things he would do is he would encourage people to come up on stage and punch him in the stomach as hard as they could. And he would laugh at them and have the crowd make fun of them and stuff like that. And they would do this for a while, and then the next act would be ready. Well, he was doing this, and one day he was leaving a bar, and as he was leaving actually entering a bar, these 2 college students who had been at the show the night before saw him, and without saying anything, they punched him 3 times in the stomach. So what was the difference between walking into the bar and being on stage? On stage, he was ready. He was able to engage those muscles, get his core muscles, tighten them up. He wasn't able to do that because they punched him unexpectedly. They ended up rupturing his spleen, and he died 3 days later. So that ability to protect our internal organs is a vital function of muscle tissue. So there you go. Let's see if we got it all. Producing body movements, stabilizing body position, moving substances within the body by changing the volume of the organ, supporting our soft tissue and joints, maintaining body temperature, nutrient reserve. Excellent. Perfect. We've got it all. And what's amazing about this is muscle is pretty much a one-trick pony. Muscle tissue, and let's be specific, does one thing and one thing only. What is that one thing really that muscle tissue does?
Contracts.
Contracts. Exactly, absolutely. It pretty much does one thing, it contracts. All right, so now we know the functions of muscle tissues. And again, let's go back. Remember, at this point, we are talking about all muscle tissues. Someone remind me again how many muscle tissues there are?
600?
Not how many muscles, named muscles, how many muscle tissues? There you go. There are three muscle tissues. And what are they?
Smooth muscle.
Smooth.
Cardiac.
Cardiac.
And skeletal.
Skeletal. Excellent. Perfect. Excellent. Absolutely. So again, when we talk about these functions, these are functions of all of the types, smooth, cardiac, and skeletal. All of them are capable of doing that. And I make a point of emphasizing that because when we talk about the properties of all muscle tissues, we're again talking about all muscle tissues. All right, smooth, cardiac, and skeletal. All right, so 4 main properties, or again, we can use the term characteristics of muscle tissue. Again, just like epithelial tissues were superficial. They were avascular. They were cell dense. Connective tissues, right? Their cardinal characteristic is they have a matrix of some sort, right? Well, there's 4 main characteristics that all muscle tissues share. The first is that it is irritable. What does it mean to be irritable?
Can be irritated.
Okay, true. The building is tall because it is tall. What does it mean to be irritated?
Easily excitable or worked up.
I like that. And it doesn't even necessarily have to be easily, but you have the right idea. What it means to be irritated is basically to be able to receive a stimulus and respond to it. Right? Giants fans tend to get that way this time of year, right? Every time the Dodgers plunk down another $100 million to get yet another big player, right? Where the Giants aren't able to do so, right? They get very irritated. Of course, however, when I ever think of being irritated, I always think of being irritable. I think of in terms of my grandmother. My grandmother was the most sweet, loving, caring individual in the whole wide world, except when her stories were on. You did not interrupt grandma while she was watching her stories. Right? When those soap operas are on, they had 100% of her attention. And if you did so much as talk, she would take off her shoe and throw it at you. Right? You did not disturb grandma during her stories. And that's what it means to be irritable, to receive a stimulus and respond to it. Now, in particular, muscle tissue responds by producing an electrical signal. So because it responds by producing an electrical signal, we say it is electrically excitable. So we can say it either of those 2 ways. It is irritable, it receives a stimulus and responds to it, or we can say it's electrically excitable. It is going to respond by producing an electrical signal, a big positive electrical signal. Anybody know the fancy name we give to that big positive electrical signal?
Action potential.
Exactly. Awesome. We call it an action potential. Excellent. Nervous tissue does the exact same thing. When we talk about the nervous system, we'll talk about neurons that are also irritable. They're also electrically excitable. When they get stimulated, they also produce a big positive signal called an action potential. So there you go. It's irritable. It can receive a stimulus and responds to that stimulus, and it produces a big electrical signal that we call an action potential. Excellent. Now, when it produces that electrical signal, when it produces that action potential, it causes the tissue to have contractility. This is an active process. When I say active process, what do I mean by that?
Requires energy.
Yeah, absolutely. It uses energy. And of course, that energy is going to be in the form of what?
ATP.
Excellent. Excellent. And what does it mean to contract?
To shorten.
Absolutely, right. What this means is that it is ability to change its length when stimulated. This change in length generates tension. Or again, if we want to think of it in terms of physics, it generates a force, allowing it to do work. Move things through our body, move our body through space, hold our organs in place. And because that generation of force isn't 100% efficient, some of that is lost in the terms of heat. All right, we comfortable with that?
Yep.
I make a point of emphasizing this because this is very different from its third property or characteristic, extensibility. What is extensibility? Huh. Anyone know what I mean when I say extensibility?
Ability to lengthen.
Excellent, absolutely. So that is part of it, ability to lengthen, right, or ability to be stretched, but without damage, right? Now, I don't have a rubber band, but I have one of these fun little stress things, right? This is a stress thing that can be stretched out, and it can be stretched out without damage. Now, are there limits to how far I can stretch it without damaging it? Yeah, absolutely. But here's the other key as well. If I just sit this on the counter and watch it, is it ever going to stretch out on its own?
No.
No. And that's the key. Extensibility is actually a passive process of this, of muscle tissue. It does not use ATP. And this is not, all right, someone's making a little too much noise making breakfast, so I'm going to go ahead and mute everybody real quick. Excellent. All right. It does not use ATP for this process. It's basically something that happens to. This extensibility is the ability to be stretched without damage. Again, there are limits to how far it can be stretched. But again, ability to be stretched. It doesn't stretch itself out. Notice what that means. When I am, and now we know the correct term for this, decreasing the angle of my elbow. And what's that process of decreasing the angle of my elbow again?
Flexion.
Flexion.
Flex. Excellent. So when I flex this, it is because this muscle, the superficial muscle here of my body, what's the superficial muscle of this body? Anybody know?
Bicep.
Bicep brachia. The bicep brachia contracts and gets smaller. And as it gets smaller, it pulls my forearm forward. But I also have the ability to extend my elbow. Do I extend my elbow by having the bicep brachia push the arm away?
No.
No, instead I have another muscle down here, the triceps brachia. It contracts and gets smaller. And when it does that, my elbow extends. Notice when my elbow extends, my bicep brachia is getting longer. Not because it's using ATP, not because it's presenting any force, but it is being stretched out by the other muscle. And is it damaged by that?
No.
No. So again, just like a rubber band, just like this, whatever this thing is, right? You can stretch it out and it doesn't get damaged. And here's the other thing. What happens when I let go of it?
We'll snap back together.
Yeah, it goes back to its original shape. And that's the fourth characteristic, the fourth property of muscle tissues, elasticity. Elasticity, notice, is elasticity a passive or an active process?
Passive.
Yeah, this is another passive process. Right, does not use ATP. It's something that happens to the tissue. Remember, we said muscle's just a one-trick pony. It really only does one thing. It contracts. But it has other properties, important properties. And again, one of those is an elasticity. When that tension is removed from the muscle, just like a rubber band. If I take this and squeeze it up into a ball and let it go, it goes back to its original shape. If I stretch it out and let go, it goes back to its original shape. Muscle tissue does the exact same thing. When you remove the tension from it, it has the ability to go back to its normal length and shape. And it just so happens that when we are in anatomical position, most, not all, but most of our skeletal muscle is at its normal length and shape, which is pretty darn convenient. All right. So again, these are characteristics of all muscle tissues. Questions on this? All right. So as we've talked about, these are both the functions of all muscle tissues and now the characteristics of all muscle tissues. But as we've also talked about, we know that muscle tissue comes in 3 flavors. And what were the 3 types of muscle tissue again?
Skeletal.
Yeah.
Smooth.
And cardiac.
And cardiac. Excellent. Excellent. So just like we talked about characteristics of all epithelial tissues, and then we talked about the specific types, and then we talked about characteristics of all connective tissues, and then the specific types, we're doing the same thing with muscle tissue. We've talked about characteristics of all muscle tissues, but now we're going to talk about the 3 specific types. Starting first with skeletal muscle. What would you say is the location of skeletal muscle?
Near bone.
Actually, I like that a lot. Actually, we can be even more precise than that. Not just near bone, but
Along.
attached to bone. Now, are all skeletal muscles attached to bone?
No.
No. So we do have to throw in that pesky word, most. Most are attached to bone, but there are actually some, the small ones, especially like in the facial region, where you have small muscles that attach muscle to muscle or even muscle to skin. So we can have stuff like that. Excellent. Skeletal muscle is by far the largest of our muscle tissue types. Some of the skeletal muscle cells can be as much as a foot long. You can have a single skeletal muscle cell that can be up to 12 inches in length. They also have a cylinder shape. So basically they are these long, and I'll cheat here and do this. Long cylinder shaped cells. But there are a couple other characteristics to our skeletal muscle. The first is that they are striated. What does that mean?
They have lines.
Yeah, absolutely. It has stripes. One of the things that we will see about skeletal muscle is that it has these stripes that run basically perpendicular to the length of the cylinder. The reason for this is it has some incredibly precise arrangement of proteins. This precise arrangement of proteins is more dense in some areas, less dense in others, and because of that, and they're so precisely arranged, that it actually gives the muscle cell the appearance of stripes. Now, not surprisingly, if it is chock filled with proteins, we need to be able to make lots of proteins. And to make lots of proteins, we need lots of instruction manuals. So it turns out skeletal muscle is multi-nucleated. Right? What does that mean?
Has more than one nucleus.
It has more than one nucleus. And what's interesting about the arrangement of these nuclei is because this cell is so chock filled with proteins, the nuclei get pushed towards the outside of the cell. Now, again, they are not outside the cell. They are contained within the cell, but they get pushed to the edge of the plasma membrane. So basically, they kind of bulge out on the side like this because they're pushed away by that arrangement of proteins. Kind of like what we saw in the adipocytes. Remember in the adipocytes, the nucleus got shoved to the side by that big fat vacuole. And so that's what we see here. Massive amount of proteins, so not surprisingly, a massive amount of nuclei to be able to make those proteins. Excellent. So striated, multi-nucleated, right? Big cells, cylinder shape. Most of them are attached to muscles. And then remember, when it comes to healing, there are two methods we can use to repair tissue. What were those two methods we could use to repair tissue? If only this was a cumulative class where I had to remember the stuff I learned a couple weeks ago. Well, when I have a tissue that needs to be repaired, I can either replace it with the same type of tissue, or I can replace it with a fibrous connective tissue. What do I call it when I replace it with the fibrous connective tissue? Forms the scar tissue, because I'm replacing it with that fibrous connective tissue. Fibrosis. Excellent. And what do I call it when I replace it with the same type of tissue? Come on, I know you know this one. I'll wait. I'll play with my stretchy thing. What do we call it when we replace tissue with the same type of tissue? Give you a hint. Starts with regeneration.
Replacement tissue or
How about regeneration?
regeneration?
There you go. Exactly. Excellent. And again, what do you know about skeletal tissue? Does skeletal tissue regenerate?
Yes.
To an extent.
To an extent, yeah.
If I'm doing those curls for the girls and I end up pulling a muscle, am I going to be able to repair that after a little bit of injury from that? Sure. But if I have a shark come and take out a big chunk of my shoulder, am I going to be able to regenerate all the muscle tissue that is lost as a result of that?
No.
No. So notice Yeah, notice skeletal tissue has a limited regeneration. Excellent. All right. There you go. That is skeletal tissue. Let's talk about smooth muscle tissue. So again, let's go ahead down that. Skeletal muscle tissue, smooth muscle tissue. Let's do the same thing. Where is smooth muscle tissue located?
Organs, like digestive system.
Yeah. What else? You got the right idea. It lines the walls of hollow organs. Does all smooth muscle line the walls of hollow organs? Did we learn about any smooth muscle in this class that wasn't found in the wall of hollow organs?
Yes.
Yeah, where? Where was the first time we talked about smooth muscle in this class? Maybe in the first organ system we discussed.
The inside of our mouths.
True, that is definitely one of the places that you would find it, absolutely. But you could argue your mouth is a hollow organ, so that's not quite, doesn't meet our characteristics. What was the first organ system we learned about in this class?
Skin.
Skin. Where did we have smooth muscle in the skin? Did we have smooth muscle in the skin? Look down at your arm. Get very scared. And tell me what happens.
The auricular.
There you go. The rector pili muscles.
Pili.
Absolutely. There you go. Excellent. Look, I appreciate we are on the verge of a 3-day weekend, guys, but come on. Let's not check out quite yet. Most smooth muscle lines the walls of hollow organs. All right. Let's be consistent. Smooth muscle, if skeletal muscle are the largest cell types, smooth muscle are the smallest cell types. These are typically arranged in sheets, layers of cells that work together. They have what we call a spindle shape. This spindle shape basically has a large centrally located body and then two tails that taper off from this. Now, obviously, this is not drawn to scale with the skeletal muscle one that's next to it, but we're looking at the size here. And of course, it's called smooth muscle because unlike skeletal muscle, it is non-striated. No stripes. Meaning that it doesn't have the same proteins arranged in the same way that our skeletal muscle does. So when we learn how a skeletal muscle cell contracts, we're going to see that smooth muscle doesn't use that same method. Now, it doesn't mean it doesn't have a lot of proteins. It does have a lot of proteins, but it is only uninucleated. What does that mean?
One nucleus.
One nucleus, exactly. Just one big centrally located nucleus. Oh, I know what I forgot to say about these things. And when it comes to healing, what do you think about smooth muscle? Do you think it regenerates better than skeletal or worse than skeletal?
Better.
Yeah. In fact, it has the highest rate of regeneration of all of our muscle tissues.
Would that be because it's not it doesn't contract like skeletal muscles, so therefore the injuries wouldn't be as extensive?
I don't know about the extensiveness, but you have the right idea. The as we'll see, how you make a skeletal muscle cell is something that's really tricky. And so, yes, being able to make more of them is something that is very tricky as well because of that. This is a simpler cell shape, so not surprisingly, it is easier for us to make more of them and to arrange them in the sheets. Absolutely. Excellent. There is one other big, huge difference between skeletal muscle and smooth muscle. And what is it?
It is passive.
What do you mean by passive?
Like when you go to flex your arm, that's an action you have to take. You don't think about using
It's
your
involved.
smooth muscles. It's just involuntary. It just happens.
Yeah, there you go. Exactly. Absolutely. How we control them. You guys are absolutely right, right? Samuel's got it as well. Absolutely. How we control them. As you mentioned, skeletal muscle is something that we voluntarily control. So it is something that, voluntary control, something that we make that conscious decision. And so what happens is it is controlled by our somatic motor nervous system. Right? If I want to move my hand, I make that decision as we talked about on the contralateral side of my brain and send a signal to it and move. Right? If I lop my hand off and put it in a petri dish and provide it with all the oxygen and nutrients that it needs, is it ever going to wave its fingers on its own?
No.
No. All right. What about my stomach? Can I voluntarily say, all right, stomach, contract?
No.
No. So it has involuntary control. But if I rip my stomach out, I put it in a petri dish and I provide it with all the oxygen and nutrients that it needs, will it contract on its own?
No.
No. So while it is involuntary, I don't know why I have a problem spelling voluntary, involuntary control, it is controlled by what we call our autonomic motor nervous system. Right? Sympathetic and parasympathetic, rest and digest, a fight and flight. Is there anything else that can control smooth muscle? Well, I put an "and" there, so there must be something.
Yeah.
Do we have another organ system that is involved in communication and control?
Endocrine.
Endocrine. Bingo. So it can be controlled by our autonomic motor nervous system and by hormones. And that brings us to our cardiac muscle tissue. What is the location of our cardiac muscle tissue?
Heart.
And anywhere else? Is it mostly heart, like mostly attached to bones, or mostly in the hollow organs? No, all heart all the time, exactly. Only in the heart. Now, not surprisingly, if skeletal muscle is the largest and smooth muscle is the smallest, this is going to be intermediate in shape, in size. Whoops. When it comes to shape, it has what we call a branching shape. It is similar to skeletal muscle in that they're kind of cylinder-shaped cells, but these cylinder-shaped cells will have multiple branches coming off of them. So they have this kind of irregular branching shape. They're kind of this interesting combination of skeletal and smooth. Cardiac muscle is striated, meaning that it has stripes. That same arrangement of proteins in it that skeletal muscle has, meaning it is going to use a very similar method of contraction to what skeletal muscle does. But like smooth muscle, it is also uninucleated. So it has a big, large, centrally located nucleus. Yes, there are a few cells that can have two. Very few, but occasionally you say, oh, binucleated. But most of the cardiac muscle cells you're going to look into are uninucleated. So I mention it because your book mentions it, but you don't have to worry about that. And let's talk about our last two characteristics. When it comes to healing, how much regeneration does cardiac muscle do?
None.
None. No regeneration. Right? When that cardiac muscle tissue gets damaged, it cannot repair itself, which is why damaging your heart is so dangerous. Now, when it comes to control, voluntary or involuntary?
Involuntary.
Absolutely. But remember, I cut my hand off, provide it with oxygen, nutrients, it sits there. Rip my stomach out, put it in a petri dish, provide it with oxygen, nutrients, it just sits there. If I rip your heart out, put it in a petri dish, and provide it with all the oxygen and nutrients that it needs, is it going to contract?
No.
Turns out, yes.
Well, you'd
And
have to
not
supply.
only,
You'd have to supply it with electrical.
no, you don't have to supply it with electrical. That's the amazing thing about cardiac muscle. If I take that cardiac muscle out of your body, provide it with all the oxygen and nutrients that it needs, it can actually produce its own action potentials. We have a fancy name for that. We say that it is autorhythmic. In fact, if I rip your heart out, put it in a petri dish, provide it with all the oxygen and nutrients that it needs, it will beat at a rate of about 100 beats per minute. Is that what your heart rate is beating right now at?
No.
No, unless you had a quad shot from Dutch Brothers before you got here today. It probably isn't going 100 beats per minute, right? So what's cool about cardiac muscle is that autorhythmic. It is able to stimulate itself, produce its own action potentials. It doesn't have to be stimulated by other systems, but it can be modified. Right? Like I said, while you're sitting here calmly in the classroom, you have a certain heart rate. When you're exercising after class today, you have a certain heart rate. Right? When you're meditating, you have a certain heart rate. Our heart rate can go up and down depending on our needs, depending on our emotions, depending on lots of things. So it can be modified. And it's modified by the autonomic nervous system. Oops, I don't need this all to be captified. So it can be modified by our autonomic nervous system and by hormones, but it's not. But it actually is able to produce its own action potentials. All right, let's put this all together. We've done it here with all the pretty words. Let's look at all the pretty pictures. Skeletal muscle. Where do we say it was located again?
Most
Touched.
bones.
Right, mostly connected to bones, absolutely. We know it is striated, meaning it has stripes. We know it is voluntary, controlled by our somatic motor nervous system. It is multi-nucleated. Smooth muscle is located where?
Lining our hollow organs.
Most lining the hollow
Most.
organs, right? Non-striated, involuntary, controlled by hormones in the autonomic nervous system. And our cardiac muscle, where was it located?
The heart only.
Yep, only in the heart. Striated, involuntary, uninucleated, and again, unlike the other cells, autorhythmic. actually able to produce its own action potentials, but it can be influenced, it can be modified by hormones and our autonomic nervous system.
I had a question.
Yes.
I think we mentioned it a while back, but what gives cardiac muscle tissue its ability to contract at the same time? Was it because it had gap junctions?
Yes, absolutely. So as we will see, when it comes to, right now we're talking about just the single cell. When we want the heart to work together as a continuous pump, all the cells are connected together by gap junctions, so that when one cell gets stimulated and starts to contract, that stimulation is spread to the next and spread to the next and spread to the next and spread to the next. So basically all of them are going to work together as a single unit. Exactly. And here we have some pretty pictures. Here we see our skeletal muscle. Notice a couple things about this. And again, there should be some things that you can easily see. So let's start here. Notice there is definitely a, whoops, that's too big. A lighter region that we can see here, and then a darker region that we can see next to it. Notice inside of the light region is a dark line down the center of it. But also, if you look really closely, in the center of the dark stripe is an area where it's a little lighter in color as well. These variations of color, notice, go through the entire cell. We are looking at just parts of 1, 2, 3, 4 skeletal muscle cells at a very high magnification. So this is just a small part of a single cell that goes well beyond here. Notice we can see the nuclei are pushed to the side by all that precise arrangement of proteins. But notice one other thing as well. Not only are these proteins perfectly aligned within this cell, but they're also perfectly aligned with the cell next to them as well. There is tremendous precision in the organization of this muscle tissue, of these proteins. So my question becomes, why? Why do we need this precision?
Because they need to contract at the same time in order to work together.
Exactly. That precision gives us tremendous coordination and tremendous strength, right? I could take any 4 of you and put you in a boat and give you some paddles. And could you get from one side of Lake Folsom to the other side of Folsom Lake?
Yes.
Absolutely. However, if I made you compete against a crew team, and assuming it's not Lori Lofton's kids, what are the chances of you guys beating that crew team?
Ain't happening.
Yeah, not very good, right? Not because you guys aren't tremendously fit and in shape, but that crew team has been working together. And one of the primary things that they've been learning is precision. They are all putting their oars in at the same time and pulling at the same time and raising and rotating them at the same time. And that precision enhances the effectiveness of their movements. So this precision to the organization and to the function of these muscle cells are going to allow them to generate a massive amount of force, a massive amount of tension. All right, and we can summarize the characteristics. What was the shape of our skeletal muscle? Cells?
Cylinder,
Cylinder. Where do we find them?
mostly attached to the bone.
Excellent. Voluntary, yes or no?
Yes. Voluntary.
Yes. Striated, yes or no?
Yeah.
Yep.
Excellent. Number of nuclei, 1 or many?
Multi-nucleated.
Multi, excellent. Able to regenerate?
Yes.
Yeah, limited. Limited, absolutely. Notice here, we see our smooth muscle. Now notice, remember, as we said, these arrange into layers. They have that spindle shapes, but the spindle shape kind of overlaps with each other in a way. When you're looking at it in a single sheet, it is hard to see the individual cells. But here we see a layer of smooth muscle. And I just gave it to you, but what is the shape of smooth muscle?
Layered.
Well, again, the shape of the individual cells are spindle shape, right? They have a large centrally located nucleus and then the tapered ends to them. You don't necessarily see that spindle shape when they're all together into a single layer, but each individual cell has that spindle shape. Where do we find our smooth muscle?
Lining the hollow organs.
Mostly lining
Mostly.
the wall of our hollow organs. Excellent. Voluntary, yes or no?
No.
Striated, yes or no?
No.
Number of nuclei, one or many?
One.
One. Excellent. And able to regenerate?
Yes.
Most efficient.
Yeah, most, yeah, absolutely. Most efficient of all of them. And then lastly, cardiac muscle. What's the shape of cardiac muscle?
Branching shape.
Branching shape. Absolutely excellent. One of the, where's it found?
Heart only.
Heart only. Voluntary?
Involuntary.
Involuntary, so no. Striated?
Yes.
Yes, absolutely. Number of nuclei, one or many?
Mostly one, sometimes
Mostly
not.
one, yeah. Every once in a while, you might fail a second one, but mostly one, absolutely. In fact, what I want to show you about this tissue that we're looking at here is a couple of things. We'll talk about the histology a little bit more at the end of class today, but these nuclei that you're seeing right now are not actually the nuclei of the cardiac muscle cell. Notice these cardiac muscle cells, as we mentioned, because they have this branching shape to them, there's a lot of space in between. And this space in between is filled with connective tissue. And so these big nuclei that you see are the actual nuclei of the connective tissue in between. They kind of form like a connective tissue skeleton inside of this cardiac muscle. Notice also, we can see that it is striated like we talked about. But notice one other thing as well. There are the striations as we talked about, but notice also there are these much darker lines as well. These darker lines are actually what are known as the intercalated discs. These intercalated discs are actually where the cardiac muscle cells come together. And as Jacob pointed out earlier, in these intercalated discs are where we find a massive number of cell junctions. Two in particular. The first are desmosomes. What did the desmosomes do for us again?
Desmosomes are the anchors.
Yeah, they're those spot welds that really hold the cells together really, really tightly, because our heart is generating a massive amount of force, and so we need to hold the cells together tightly to do that. But also, as Jacob pointed out, this is where we find the gap junctions. Those gap junctions basically work to allow the cells to rapidly communicate with each other so that they basically work together as one. So when one fires an action potential, it spreads to the next, and it spreads to the next, and the whole tissue contracts together as one, forming this huge, very efficient pump to pump our blood through our body. Able to regenerate?
No.
Nope. And again, as we talked about, this is the one that not only is it involuntary, but also autorhythmic, able to generate its own action potentials. All right. Questions on that? All right. Again, notice up to now, we have been focused on all types of muscle tissue. But now our focus is going to change. Now we are going to solely focus on skeletal muscle tissue. You are going to learn everything you ever wanted to know and more about skeletal muscle tissue. And then on pretty much the last day of lecture, we will finally come back and compare all that we have learned very superficially with smooth and cardiac. We'll get much more into cardiac muscle, much more into smooth muscle in 431. When we talk about things like the digestive system, when we talk about things like the cardiovascular system, excuse me. So for the rest of this section, almost all of the rest of the section, we are going to focus on skeletal muscle tissue. And then on the very last day, we'll take all that we've learned about skeletal muscle tissue and briefly compare it to smooth and cardiac. All right, questions on that? All right. So what we are going to, our goal for this next section, like I said, is to focus on skeletal muscle tissue. Because ultimately we want to get to the skeletal muscle system and those 600 named muscles of our body. And we want to understand how a muscle contracts. We are going to learn that entire process. It is the longest physiological process you will study probably in all of anatomy and physiology. The good news is it's not hard, but it is long. We have to go from everywhere where I decide to contract the muscle to the point where that muscle is generating a force and moving our body through space. But to truly understand how that physiology works, we need to understand the anatomy. Not just the anatomy of individual muscle cells, of individual muscles, like the 600 named muscles of the body, but the anatomy and understanding of an individual muscle cell and all those proteins inside of it. So we have a massive amount of anatomy and a massive amount of vocabulary in front of us. And because of that, I want to make sure we're coming at this fresh. So we're going to take our first break. We'll take a 15-minute break. Looks like it's a 9:10 right now, so we will restart at 9:25 and be prepared for a massive amount of anatomy and vocabulary. And I will start the recording at this time. All right. Questions on that? All right, I will see you guys in 15 minutes. All righty. So let's go ahead and get started. As I mentioned, our goal is to understand how our muscle tissue contracts. And to do that, we have to break it down. We are going to start with one of those 600 named muscles in the body, like that bicep brachia. That muscle is obviously made up of hundreds, if not thousands, of muscle cells, all put together. And as we've talked about, muscle cells are these long cylinder-shaped, thin structures. And because of that, they use the term muscle cell and muscle fiber interchangeably. And we'll do the same thing. But obviously, to make a muscle like the bicep brachia, we can't just have thousands of muscle cells that you throw together in a bucket and call it done. They are going to need to be arranged and organized. And we arrange them and organize them by many layers of connective tissue. Now, when we talk about this connective tissue that organizes and arranges these muscle cells, this is not a concept we're not unfamiliar with. The first is that individual muscle cells will have, or groups of muscle cells will have connective tissue fascias that wrap around them and distinguish them. Now, again, not a new concept, not something that you aren't aware of, because now that the world is open back up again, so this weekend being the 4th of July, perfect thing to do would be to go to a Renaissance fair. And of course, the whole reason to go to a Renaissance fair is to get that big, huge, massive turkey leg. Now, when you get that big, huge, massive turkey leg, is the turkey meat on that turkey leg just this big, huge, amorphous circle of meat that is around that bone?
No.
No, notice as you start biting into it, right, or tearing at it with your fingers, individual muscles come off. Now, the reason those individual muscles come off is when it was cooked, the connective tissue, the collagen that was holding those individual muscles together is broken down. So for instance, in my arm, I have some muscles that are going to be involved in flexing my elbow and some that are going to be involved in extending my elbow. And I want to separate those groups from each other so they can work independently of each other. So we're going to have all these fascias that are going to help to distinguish and define them. Those individual fascias are what we know as the deep fascias. They're made up of dense irregular connective tissues. And like I said, they stabilize and hold muscles in place. But if you remember, we've actually already talked about another fascia. Of course, we didn't use the term fascia when we were talking about it. We used two other terms. One was the subcutaneous layer, and the other was the hypodermis. Well, the subcutaneous layer, the hypodermis, has a third name that we can give it, and it is that superficial fascia. Remember, as we talked about, our skin is only loosely connected to our muscles, so that as we contract our muscles, those muscles can change shape underneath the skin and move more freely without putting too much tear, too much tension on the skin itself. That, remember, is primarily areolar and adipose tissue. So we have that areolar and connective tissue and adipose that is acting as that superficial fascia, but also individual muscles have that deep fascia that wraps around them and holds them in place. All right. Questions on that? All right. From there, then, we can look at an individual muscle. Again, this is the organ that is a muscle. And as we can see here, it is made up of hundreds, if not thousands, of individual muscle cells. Here is a single muscle cell. But as we can also see, there's a tremendous arrangement of these muscle cells. So let's talk about that arrangement of these muscle cells. Starting first with, again, when we talk about a single, and I think I need to make this a little bit smaller, a single muscle cell. Remember, one of the things that we talked about with a single muscle cell is that that single muscle cell is irritable. It is electrically excitable. What does it mean to be irritable? What does it mean to be electrically excitable again?
Able to be contracted with electrical current stimulus.
Right, absolutely. It produces, when stimulated, it produces that electrical signal, that action potential. And it is actually specialized to not just produce an action potential, but spread it along that entire muscle cell. Kind of like a wire in your wall. The wires in your wall can have electrical signals that spread along them. And so if you peeled open your wall, would you see a bunch of just copper cable wires just intermeshed with each other? No, because the problem would be that the signal might jump from one wire to another, and we don't want that. So each individual muscle cell, if you look at this illustration, has a little bit of connective tissue that wraps around and insulates each individual muscle cell. So each individual muscle cell is surrounded by an areolar connective tissue. Now, this certainly isn't the only place we find areolar connective tissue in the body. So not surprisingly, this areolar connective tissue, we give a name, and hopefully I've used the term based on location enough in this class that I can just abbreviate it BOL. And this areolar, oops, if I spell areolar correctly. There we go. Hold on. There we go. The areolar connective tissue, based on its location, we call an endomysium, because it is within the muscle, surrounding each individual muscle cell. So each individual muscle cell has its own little insulation that wraps around it, that areolar connective tissue that, based on its location, we call an endomysium. However, notice this muscle cell and 14 of its closest friends are bundled together. And we bundle them together into a structure we call a fascicle. So notice here, and I'll change colors. We have a connective tissue that is wrapping this muscle cell and 14 of its closest friends into a single structure known as a fascicle. This fascicle is formed by another connective tissue, and this one happens to be a dense, irregular connective tissue. And this certainly isn't the only dense, irregular connective tissue we find in the body. So this one, based on its location, we call it a perimysium. And then when we look at the muscle itself, we see that it isn't just made up of one fascicle. It is made up of dozens, if not hundreds, of fascicles. And so all these fascicles are bundled together. And not only are they bundled together, but also they're bundled together with the blood vessels and nerves. And we bundle these fascicles with the blood vessels and the nerves, and we do that to form the muscle organ. So notice here on the outer surface, we have yet one more connective tissue that bundles all of these things together to form our individual muscle, that muscle that is the organ. Now, of course, we need a connective tissue to bundle these up into a muscle, and it is, again, a dense, since I wrote it out once, I'll already do a dense, irregular connective tissue that forms the muscle. And of course, we have to give it a name based on its location, and based on its location, we call it an epimysium. Now, you may not have thought of it in these terms before, but I know you're aware of it. If you want, at some point later in this class, we can have the deep philosophical discussion about whether or not meat is murder, right? If you're a staunch vegetarian or vegan, right, you may be very, very much opposed to the consumption of meat for fuel. However, and again, we can have that philosophical debate, but what we can't debate is that meat is muscle, right? And again, you may not have ever thought about these fascicles, thought about these perimysiums, thought about the arrangement of these muscle cells ever before, but you might be aware of it. Because let's say, as we mentioned, the 4th of July is right around the corner. So maybe you decided to splurge, and so you bought a really nice, expensive tri-tip at Costco. And for the past 3 days, it's been marinating in the bottom of your refrigerator. And so come the 4th of July, you're going to bring it out, pat it off, bring it up to room temperature, get it onto that sizzling hot grill to get that nice crust on it, and then low and slow, bring it up to the perfect temperature. Take it off, let it rest for 25 minutes to reabsorb all those juices and all that love and all that time and all that effort. After you do all of that, are you just randomly going to close your eyes and just cut that tri-tip however the heck you want, just randomly?
No,
No, how do you cut a tri-tip?
with the grain.
With the grain or against the grain?
Against it.
Against it. You cut it against the grain, right? Because that way you get more tender pieces. But more importantly, what the heck is the grain? Well, guess what? The grain are the fascicles. That's why you cut it against the grain. You are breaking up the connective tissue of those fascicles when you cut it against the grain, and that makes for a more tender piece of meat. Or maybe you don't want to put that time and effort into it. So instead of buying a really expensive tri-tip, you bought a really cheap pork shoulder. And instead you put it in the slow cooker and you poured a beer over the top and you waited 12 hours. And 12 hours later, when you come back to that pork shoulder, what's happened to it magically over that time?
Separated.
Yeah, it just falls apart. But when it falls apart, does it fall apart into just irregular shaped pieces? No, what happens to it?
All the fascicles kind of separate on their own.
Exactly. What happens is by cooking it low and slow, you've broken down the collagen in that perimysium. You've broken down the fascicles. And so as a result of it, those fascicles just shred. You have those long shred-like, thread-like fascicles that are broken down. You've broken down all the collagen fibers, and so it just falls apart into those thread-like pieces. Right. So again, you may not have ever thought of muscle as having this organization, but if you've ever put any time into cooking it, you are aware of these things because that's how you prep and prepare them. All right. Now, there's one other thing that I want to emphasize as well, and let's go here to do this. Again, we are talking about the arrangement of a skeletal muscle. And again, we're talking about the organ because we're working our way down. Oops. So as we talked about, we are going to have, and we'll go ahead and use black for them, individual thread-like muscle cells. Here, I'll make 4 of them. And as we talked about, these individual muscle cells are going to have that areolar connective tissue that wraps around and insulates each individual one. So again, we have muscle cells, and those muscle cells are surrounded by areolar connective tissue. That, based on location, we call the endomysium. Those muscle cells, oops, hold on, are bundled together into a structure called a fascicle. And this is done by a dense irregular connective tissue that, based on location, we call a perimysium. And then many fascicles along with our blood vessels and nerves, are all bundled together into a muscle by another dense irregular connective tissue that, based on location, we call the epimysium. And these run the entire length of the muscle cells. So we said some of these muscle cells can be as much as a foot long. But irregular connective tissues, dense irregular connective tissues, those also have collagen fibers in them. And as it turns out, the collagen fibers from the epimysium will extend out beyond the length of the muscle cells. The collagen fibers of the dense irregular connective tissues forming the perimysiums will extend out beyond the muscle cells. And even some of the collagen fibers from the areolar connective tissues that are forming the endomysiums wrapping around individual muscle cells will all extend out beyond the length of the muscle cells as well. Notice all of these collagen fibers that extend beyond muscle cells kind of all become fairly parallel in their orientation. One might even argue they form very regularly arranged collagen fibers, forming a dense, regular connective tissue. And what do you think the function of this dense, regular connective tissue extending off from the muscle cells might be?
Tendons connect to the
Exactly.
bone.
Excellent. It forms the tendons. What do those tendons do?
Attach muscle to bone.
Exactly. They form the tendons, and I'll just stick this up here so it's out of the way. All those collagen fibers that come out are going to form the tendon. That are going to connect this muscle to the bone. Excellent. Questions on this? Again, if this doesn't make sense, it is just going to get a whole lot worse from here. So if this isn't making sense, please let me know now. Here, let's take one more look at this. In a cross section. And notice this time we'll break our way down. Notice here we have a muscle. This muscle has a dense irregular connective tissue that defines the muscle. That dense irregular connective tissue, based on its location, we call an epimysium. And notice this one inside contains 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 fascicles, as well as some blood vessels and nerves. But notice we can pull one of these fascicles out because it has a lot of organization to it. If we pull one of these fascicles out, we see that that fascicle is formed by a dense irregular connective tissue that bundles individual muscle cells. And in this case, 1, 2, 3, 4, 5, 6, 7, 8 muscle cells happen to be inside of this fascicle. But notice as we look at each individual muscle cell, we see that they also have a connective tissue that wraps around, isolating and insulating each individual muscle cell. So if we pull out one muscle cell, we see that muscle cell has an areolar connective tissue that wraps around it, that based on its location, we call the endomysium. In fact, oh, where do I have it? No, that's skeletal. I don't care about skeletal anymore. There we go. This is what I want. I think. There we go. Perfect. Excellent. All righty. In your modules, under the lab handouts, you will see unlabeled muscle picture. No, that's all I want. Muscle classroom models. When you click on muscle classroom models, you'll see a couple things. First, you'll see the individual muscle tissues that we've already talked about. Based on what we talked about, what type of muscle tissue do you think this is a model of?
Cell, muscle cell.
What kind?
Skeletal.
Skeletal. Notice multiple nuclei, striations, long cylinder shape. Excellent. And notice not only would you be responsible for knowing this is skeletal muscle tissue, but I could also ask you for, say, 2 or 3 characteristics. Tell me some of the characteristics you know about skeletal muscle cells. Skeletal muscle tissue.
Multi-nucleated.
Multi-nucleated. What else?
Striated.
Striated. What else?
Cylindrical.
Cylinder shapes. And notice I just asked for 2, and you can tell those just by looking at it. That's awesome. Give me something you know about it that we can't see but can't tell just by looking at it.
Voluntary.
Voluntary. Limited regeneration. Mostly attached to bones. Again, there's nothing wrong with giving me the things that you can see. Perfectly acceptable. But I definitely I could ask you those types of questions, and more importantly, will ask you those types of questions on the exam. What do we have here?
Smooth muscle.
Smooth muscle. And again, give me some characteristics.
Uninucleated.
There you go.
Spindle shaped.
Spindle shape. There you go.
Involuntary.
Involuntary.
Lines, hollow organs.
Lines, hollow organs. No striations. Right. Excellent.
The best at regeneration.
Best at regeneration. And what's this?
Cardiac.
Cardiac. Striated, uninucleated. And if I'm being really clever, I could even point an arrow at something like that and ask you to identify that structure. What would that structure be?
Intercalated disc.
Excellent. And what type of cell junctions would you find a lot of in that intercalated disc?
Desmosomes.
Gap junction.
And gap junctions. Awesome. Excellent. Perfect. So those are things that I guarantee are in your lab exam pool. But notice also here, why are you not loading? There it is. Can you guys see that? Looks like it still hasn't fully loaded yet. What we have here is a single muscle cell. Notice if we look closely and carefully, we can see that there is that areolar connective tissue on the outer surface. And this here that I'll paint in orange is the plasma membrane of our muscle cell. We also have another one. So there's this one where a couple different views that we'll look at, and we'll talk about the anatomy of that later. Why is this taking so long to load? I don't care about that. Here's what I want. There we go. Ooh, do I have a better view for that one? Nope. All right, so we'll cheat. Notice here for this one, we see the plasma membrane of the outside of the cell. And if you look very closely, there's this gray tissue on the outside. On the back of this is a big, huge layer of endomysium wrapping around it. But the reason I show you this is notice when we look at a single muscle cell, there's still a whole heck of a lot going on inside of that. And so while we've done a good job up to here of looking at the organization of a skeletal muscle, that organ, what we need to do next is grab one of these muscle cells and expand out this muscle cell and see what's going on inside of it. And so we're going to dig even deeper. In fact, we're going to get all the way down to the individual proteins inside of this. And once we have learned all the individual proteins that are found inside of this, then we'll be able to understand how they work together to form our, to allow for our muscle to contract. Excellent. So let's do that. Here, again, histology. Notice we see this nice cross-section through an individual muscle. So notice with this individual muscle, we see the epimysium forming the outer boundary of the muscle, stabilizing some blood vessels and stuff in there. Here we see a fascicle formed by a perimysium. And then even in between the individual muscle cells, we can see the endomysium. But notice the muscle cell itself has a lot of stuff appearing like it's going on inside of it. And so that's what we need to do. We need to dig down to the level, and just to beat the dead horse, there's our muscle cell, there's our endomysium, our fascicles formed by the perimysium, our epimysium bundling fascicles, blood vessels, and nerves. And what's nice about this is this one shows how all their collagen fibers come out to form the tendons. And remember, when we talk about tendons, like in the abdominal region, like over our top of our skull, we have big, broad, flat tendons that we call an aponeurosis. All right, we've beaten that dead horse. Let's go inside. We've gotten down to this level where we've looked at an individual muscle cell, but now we need to see what's going on inside and even break it down to the levels of individual proteins so that we can understand how a muscle cell functions. The first thing we have to do is make a muscle cell. And it turns out one of the reasons why muscle cells regeneration is so limited is you don't make muscle cells the way you made every other cell we've talked about. Every other cell we talk about, one cell divides and you get two cells, and then those two cells live happy lives. That's not how we make muscle cells. Remember, muscle cells are both massive in size and, as we talked about, have many nuclei. The reason for that is they are made from many cells. Once again, we have a pluripotent stem cell. Remember, a pluripotent stem cell is a cell that is capable of dividing to produce many types of things. Remember, we talked about how mesenchymal cells, those mesenchymal cells can divide to produce any of our connective tissue cells. Well, here, our pluripotent stem cell is a mesodermal cell. Mesodermal cells are capable of dividing to produce many types of cells, but in this case, we want them to divide to form myoblasts. And hundreds of myoblasts fuse themselves together to make a single skeletal muscle cell. So here we see the process start. Several myoblasts forming and fusing together to form this one long cylinder-shaped multinucleated cell. That is our skeletal muscle cell or skeletal muscle fiber. However, what I also want you to notice is that not all of the myoblasts actually fuse to form the skeletal muscle cell. There are a few cells that stay independent right up next to the muscle cell. They are actually going to be within the endomysium, but are not actually a part of the muscle cell. And these special cells are what are known as satellite cells. So if a skeletal muscle cell is killed or damaged, that satellite cell that is sitting right next to it has the ability to divide, to make new myoblasts, to repair or replace the muscle cell that has been lost. However, do we have an unlimited supply of these satellite cells?
No.
No. So as we talked about, that's fine if I tear a muscle while I'm exercising, but if that shark takes out a huge chunk of my shoulder, I'm not going to be able to replace it all with muscle cells. And these muscle cells are highly specialized, so instead it'll get replaced with scar tissue, fibrous connective tissue, which doesn't look the same and doesn't function the same, and so it doesn't repair. In fact, every once in a while, our model makers get clever on us. And notice if we go back to our original muscle cell picture, notice here, as I talked about, here is the plasma membrane of our muscle cell. Here is the endomysium wrapping around it. And notice here, outside of the plasma membrane, but inside of the endomysium, is this big globular thing on the front. Guess what this big globular thing on the front is?
Satellite cell.
Yeah, here we actually see this model does a nice job of showing us that satellite cell. So we can see that satellite cell really, really nicely here on this model. In fact, notice if we, I think, that's not bad, but I think if we look at it from the top, here we go, perfect. If you look closely on this model, which you should do because I guarantee you it's going to be on the lab exam, you can see the plasma membrane of the cell goes like this. So notice that satellite cell is outside of the muscle cell. It's not part of the muscle cell, right? Again, it's nucleus. It does go around the nucleus, but that satellite cell is outside. But as we saw, it is contained within the endomysium. And so that's what we have here. Those satellite cells allow for the limited regeneration that we have of muscle cells. Now, there's another big implication to this, because I'm sure, like me, you are truly exhausted by this class. All right, we've been in here in what feels like a billion years, even though this is the beginning of like our 4th week, 5th week, I don't even know, I've lost track, right? But I'm exhausted, I'm sure you are as well. So what we could do, instead of meeting for 4.5 hours each week to talk about anatomy and physiology, we could just meet for 4.5 hours each week and work out in the gym. And if we got together and worked out in the gym for 4.5 hours every day, 4 days a week for the next 8 weeks, all of us would have big, huge, hulking muscles after that time. Our muscles would be bigger. Now, would they be bigger because we made more muscle cells?
No.
No, as it turns out, that's not how muscle cells primarily grow. Instead, what happens, actually, let's do it this way. We have a muscle cell. Nope. We have that muscle cell, that long cylinder cell. And as we talked about, that muscle cell is filled with proteins. Then I work out today. I do my arms today, and then they get tired. So I rest them because they're sore and they're tired tomorrow. And why are they sore and tired after I've worked out?
Because you tore them through the workout, you damage them.
True. But although we do want to be careful with terms like torn and damaged. Yes, we have decreased the efficiency of this muscle by damaging and breaking some of these proteins. And so what happens is when we use muscles, we damage and misalign the proteins. And so when we recover, we repair the proteins and we make more proteins. And when we make more proteins, the muscle cell gets bigger. It has more proteins, and with more proteins, it can produce more force, and we are stronger. This type of growth is what we call hypertrophy. We don't make more muscle cells. Our muscle cells get bigger. With use. And that's the key to this as well. This is the classic example of use it or lose it. Because again, we're going to work out 4.5 hours a day, 4 days a week for the next 8 weeks. But what happens if you break your arm and they put it in a cast and you have to wear that cast for 8 weeks? What happens when they take that cast off and you compare your forearms?
Atrophy.
Yeah, exactly. One is tiny by comparison. Because by the same way, remember, we talked about muscle is dynamic. Muscle is a nutrient reserve. If you don't use it, it gets broken down. And the cells get smaller. And as you mentioned, we call that process atrophy. So we typically change size of muscles by increasing the number of proteins or decreasing the number of proteins. The overall number of muscle cells typically doesn't change. We just make the muscle cells bigger or we make the muscle cells smaller. Now, does that mean it's impossible to make new muscle cells? No, because we know there is some limited regeneration. And if instead of working out 4.5 hours a day, 4 days a week for 8 weeks, we did that for 8 years, we'd be one of these big, huge, massive bodybuilder type people. And when they get to be these big, huge, massive bodybuilder type people with that extreme muscle use, is there some making of new muscle cells that takes place in that? Yeah, absolutely. But that is what we call hyperplasia, where we're adding new muscle cells. But that is not the normal way we make muscles. We make muscles, or we add to our muscles by hypertrophy, not hyperplasia. Hyperplasia is adding new muscle cells. However, right, when we talk about these bodybuilders, oh, I don't have that. Hold on a second.
Would that also be based upon the stress that you put on the muscle? Like if you're lifting heavier weights, your muscle's going to tend to get bigger as opposed to lower weights, and it's going to tone?
Yes, so absolutely. Most of these bodybuilders are doing short reps. They're doing large reps, doing powerful activities to get those really big, huge, massive muscles, like like this fella. When you get to be this size, right, obviously there's some hyperplasia taking place. This is not just growth in muscle size, this is also adding muscles. But again, most of us don't look like this. So most of us don't gain muscle mass or lose muscle mass in this fashion. Now notice I happen to have this picture, not because I'm particularly fond of this gentleman. I don't have any idea who he is. However, as you can see, one of the nice things about these bodybuilders is, especially when they're showing off like this here, they do two things. They exercise to increase blood flow to the muscles to cause the muscles to swell. And they also typically dehydrate themselves so that the skin sticks to the muscles. And the nice thing about that is it makes the muscles stand out much more readily, allowing mere mortals like you and I to be able to readily identify these muscles. And I guarantee you that part of your lab exam is going to be able to recognize the muscles in bodybuilders like this. Now, you'll notice that there is some displacement of the muscles here versus a mere mortal like you and I, but conveniently enough for you, if we go back to our Canvas site, as I'm sure you've already seen, when you look in your modules and you go down to the lab handouts, there happens to be a bodybuilder anatomy handout that does a nice job of showing you how these muscles expand and displace, allowing us to be able to recognize those. So like I guarantee there's going to be a handful, 3, 4, 5 pictures, something like that, of bodybuilders that you will be responsible for recognizing superficial muscles on those bodybuilders, because we got to have some fun here too. All right. Questions on that?
Right.
Excellent. All righty. So, single skeletal muscle cell. Let's go ahead and go back to our whiteboard and do some more drawing. We are, again, continuing to break things down. So at this point, we are breaking things down and looking at a single skeletal muscle cell. And this is a highly specialized cell. One of the primary ways that it's highly specialized is, as we mentioned, it is irritable. It is electrically excitable. It is capable of producing and propagating an action potential. Obviously, producing means to make. What does propagating mean? What does it mean to propagate? Yeah, directing or spread. Absolutely. So it can make and spread an action potential. Now, as we know, it is a cylinder-shaped cell. So let's look at a part of one of them here. And let's go ahead and put a nice big circle here. There we go. Close enough. Excellent. So here is our cell. And of course, as we know, cells have plasma membranes. However, as we mentioned, this is a highly specialized plasma membrane. And because it is a highly specialized plasma membrane that is able to produce and propagate an action potentials, as I just wrote it right above, I will just abbreviate it there. We give it a fancy name. And so the fancy name for the plasma membrane of a skeletal muscle cell is a sarcolemma. And there's one other interesting thing about the sarcolemma of our skeletal muscle cell. Yes, it covers the entire surface of our cell, but the other interesting thing that our sarcolemma does is there are large folds in the plasma membrane that penetrate down deep into the skeletal muscle cell. So there are these large invaginations of the sarcolemma that penetrate deep into the muscle cell. And of course, we need to give a name to that. So we call them the transverse tubules, which, if you're writing this out for an essay question, for instance, once you write out transverse tubules once, you can abbreviate it as the T tubules. Now, these are folds of the plasma membrane that penetrate deep into the muscle cell, but they are still connected to the outside. So these transverse tubules are filled with the interstitial fluid. Why is that important? Well, because remember, there are differences in the ion concentrations inside or outside the cell. So here in this transverse tubule, would you expect there to be a large amount of sodium or a small amount of sodium?
Large.
Large amount. A large amount of potassium or a small amount of potassium?
Small.
Small, right? Because it is interstitial fluid, it is from outside. Excellent. We also know that these cells, again, have a nucleus. We know the nucleus is going to be shoved to the side and it has many nuclei. And of course, the third component of any cell is the cytoplasm. Right? We have plasma membrane, nuclei, cytoplasm. Those were the three components of a muscle cell or of any cell. And sure enough, the cytoplasm of our muscle cell is going to be specialized. So we give it a special name, and that special name is the sarcoplasm. Yes, it has all the normal things we expect, large amount of sodium, small amount of sodium, large amount of potassium, yammer, yammer, yammer. But remember, we also talked about how there are inclusions. Inclusions are special proteins or substances that can be found inside of cells that are unique to those cells. One of those is that in these muscle cells, as we've already talked about, there is a massive amount of glycogen. What do we use that glycogen for again?
Energy used to
Yeah.
create ATP.
We're going to use it to make energy and use it to make ATP. Now, if we are going to completely and fully break down this glycogen to maximize the amount of ATP necessary, what did we say we needed? What organelle is going to help us to efficiently break down this glycogen and maximize the energy that we get from it?
Mitochondria.
Yeah. So we're going to need a massive amount of mitochondria. However, to completely break down our glucose, mitochondria needs something special. If I remember correctly, there was an equation we came up with for cellular respiration. What was it that mitochondria need to be able to fully and completely break down that glucose?
Oxygen.
oxygen. So the other thing we need to store in here is we have to have a massive amount of oxygen as well. And luckily, that comes in the form of a special protein. And that special protein is called myoglobin. Now, what does myoglobin sound an awful lot like?
Hemoglobin.
Hemoglobin. And remember, hemoglobin is the special protein that holds oxygen inside of a red blood cell. Well, this myoglobin holds oxygen in the muscle cell. So notice among the other specializations we're going to see inside of this cell is able to contain massive amount of resources, helping it to be able to make lots of ATP. All right. So far, so good?
Yep.
Excellent. Now, it has lots of nuclei. It has lots of mitochondria. The other thing that it has a lot of is the smooth endoplasmic reticulum. And not surprisingly, in our muscle cell, we give it a special name. It is called the sarcoplasmic reticulum. If you remember, we talked about how the sarcoplasmic reticulum or the smooth endoplasmic reticulum in a typical cell can help to make lipids. It can help to affect the plasma membrane of the cell. But remember, we also said how sometimes it can be specialized for storage. And that's the case here. Here, our sarcoplasmic reticulum is specialized for the storage of calcium. I'm going to draw it. It's typically this kind of mesh-like structure, but I'm going to cheat and just kind of draw it as a long, narrow line, where, again, it stores a massive amount of calcium inside of this sarcoplasmic reticulum. It is found throughout the muscle cell. But what's interesting about this sarcoplasmic reticulum is that when it gets close to the transverse tubule, it enlarges. There's a big, huge enlargement to it right next to our transverse tubule. And this big, huge enlargement is what is known as the terminal cistern. So we have this terminal cisterna, which is the enlargement of the sarcoplasmic reticulum right next to the transverse tubule. And it turns out the same thing happens on the other side as well. We have some sarcoplasmic reticulum over here and a big, huge enlargement, and a second terminal cisterna. And it turns out these three things together, the two terminal cisternae and, oops, that's not an and, the transverse tubule, form a unit called the triad. All right. I guess if I put this up here, then I can put that there. I can remind us with an arrow here. Excellent. All right, questions on this? Notice none of these have a tremendous amount of proteins, and none of these would necessarily be responsible for the stripes we see in our muscle cell. But that's because we haven't talked about that yet. There is one more very important organelle that is found within our skeletal muscle cell. These are long thread-like organelles that run the entire length of the muscle cell. And these organelles are specialized contractile organelles. These are the ones that generate the force. These are the ones that generate the tension that are going to move our body through space. These are filled with a massive number of perfectly arranged proteins. And so not only are these the functional units, but these are also the structural units, or structural organelles. These are what give the muscle cells their stripes. And the reason for this is that they're made up of a repetitive pattern of proteins. And that repetitive pattern of proteins is called a sarcomere. Now, I've thrown a lot of vocabulary at you, but again, I appreciate that my art is not the best. So let's take a look at the pretty pictures from your textbook. Here, we see the plasma membrane on the outer surface. We see the nuclei that have been pushed to the side by these massive number of thread-like organelles known as the myofibrils. Did I say that?
No.
I did not. Excellent. Let's go back to that. This big, long thread-like organelle is called a myofibril. And again, like I said, it is made up of sarcomeres. Excellent. Let's make that a little bigger. Perfect. So here we see dozens of myofibrils, a bunch of mitochondria in here, and the nuclei shoved to the side. And again, fun with vocabulary. Plasma membrane on the outer surface is the sarcolemma. It is specialized, stop it. It is specialized to produce and propagate an action potential. The sarcoplasm is all the hunk and junk on the inside. And as I mentioned, it contains some inclusions like massive amount of glycogen granules, providing the sugar, myoglobin holding the oxygen, and then a massive amount of mitochondria to take that glucose, break it down completely with the oxygen, and make a massive amount of ATP. Notice our transverse tubule has these invaginations, these folds in it that migrate deep into the plasma membrane. And these folds in the plasma membrane are what are known as the transverse tubules. They contain interstitial fluid. And notice they wrap around the myofibrils, those long thread-like organelles. But notice also associated with those transverse tubules are, here in blue, our sarcoplasmic reticulum. This specialized smooth ER where we store massive amounts of calcium. Remember, calcium makes cells do wonky things, and a muscle contraction is definitely a wonky thing. These sarcoplasmic reticulums have the second highest concentration of calcium in the body, right? The highest concentration is obviously in the matrix of our bones, but here we have the second highest concentration of calcium. And notice also right next to the transverse tubules, that sarcoplasmic reticulum enlarges to form the terminal cisternae. And those terminal cisternae, the two terminal cisternae, and the transverse tubule collectively form a unit, form a structure we call the triad. And then our last and probably most important organelle are these long thread-like myofibrils. These are the contractile organelles containing massive numbers of proteins. And of course, because these proteins are found in a muscle cell, they call them myofilaments. But it's just muscle proteins, that's all that means. And they are perfectly arranged, giving it its precision, increasing the efficiency of the muscle contraction. But that precision is also what gives the muscle cell its stripes. And that organizing structure, we see one of them right here, from here to here, is a sarcomere. I always think of this in terms of a train, right? If you're driving around Roseville, there are several locations where train tracks cross the road. And if you get stopped there, you could sit there for 5 minutes while a single train goes by. But if you think about it, a single train is not a single structure. It is hundreds of boxcars all put together. And every single one of those boxcars looks identical to the next one. So as you see that train go by, you just see boxcar, 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 boxcar as it goes by. Well, that's what a myofibril. A myofibril is just sarcomere, 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 sarcomere. A bunch of sarcomeres added together in a row to make that myofibril. Questions on this? Again, if you're getting lost or it's not making sense now, it is just going to get worse. So if this is difficult, if challenging, if you're not understanding this, now is the time to ask.
The transverse tubules, are those along the muscle cell at incremental, like individual things, like because they look like they're ordered?
No. Yes, our artist has drawn them very ordered and organized like that, but no, there isn't a precise location of the myofibril, for instance, where all the transverse tubules are located. No, it's not that precisely arranged. All right, excellent. So if we are
I did have one more question. It's more off of the topic of, but I noticed everything is, the prefix is sarco. Is that
flesh? Sarco means flesh. So yeah, because we're talking about muscle. Yeah. All right, excellent. So now we need to break this down even further. Now we are going to take one of these myofibrils, pull it out, and look at this repetitive pattern that is a sarcomere. So our goal now that we have the basic anatomy of a muscle cell is to pull out one of these myofibrils, look at this repetitive pattern that is a sarcomere, and more importantly, identify some of the proteins that form them. There are dozens of proteins that are involved in making this precise organization of the myofibril and this precise organization of the muscle cell. We are going to focus on the 7 most common proteins, or at least the most important ones, talk about the different types of proteins, and talk about how they're precisely arranged to give us our stripes. But once again, this is going to be a lot of work and a lot of vocabulary, and I want to come at this fresh. So this is the perfect opportunity to take our second break. It is 10:35, so let's restart at 10:50. And at 10:50, I will start the recording again. All right. Any questions before we take our next break? All right. Caffeinate, because we got a lot more vocabulary to go. I will see you guys in 15 minutes. All righty. Our goal now is to break down the myofibril. And as I mentioned, that myofibril is comprised of a repetitive pattern of proteins that we call a sarcomere. So what we need to do is talk about the anatomy of the sarcomere. To do that, we have to talk about proteins. So let's go back to our whiteboard. When we are talking about our proteins, there are 3 types of proteins that form our sarcomeres. Now, as I mentioned, there are literally dozens of different types of proteins. We're only going to be responsible for 7, but those 7 still fall into one of 3 categories. The first of those categories are what we call our contractile, and actually let's do it this way. Contractile proteins. And based on the name, what do you think our contractile proteins do?
Contract.
Yeah,
Or
exactly.
Aid in the process of contraction.
Yeah, well, actually, they not just aid in it, but they're the ones that are actually responsible for it. And actually, I don't like it that size. Let's go ahead and make it back bigger again. These are the proteins that actually produce the contraction. Right? These are the ones that generate the force or the tension, again, depending on how we want to think of it, that is going to cause the movement to take place, cause the change in shape. Excellent. Now, these are the ones that generate the force that allow me to move my body through space. However, notice the muscles of my arm aren't in a constant state of contraction. Why aren't they in a constant state of contraction? Would I want them to be in a constant state of contraction?
No.
No, that wouldn't be very useful. I need to be able to turn that contraction on and off. To do that, I need a second class of proteins, what are known as regulatory proteins. These regulatory proteins are what basically turn the contraction on and off. They control the contraction. Excellent. And remember to make this contraction as efficient as possible. I need that tremendous precision. I need that tremendous organization, right? And that allows the efficiency. So my third type of proteins are the structural proteins. These structural proteins are the ones that align the proteins. This forms our sarcomere, gives us the precision, and this is what causes the stripes. So, three main types of proteins: contractile, regulatory, and structural. Now, as I mentioned, there are dozens of these different types, but let's talk about these one at a time. Move that up there, move that up there. For our contractile proteins, there are two contractile proteins you are going to be responsible for. So let's do this first. Two there. Make it red. And those two proteins, the first one is myosin. Myosin is a dimer. What does that mean? What does it mean for a protein to be a dimer? Don't get me wrong. It warms the cockles of my heart when you guys don't know things about chemistry. Makes me very, very happy because chemistry is stupid. But anybody know what it means to be a dimer?
Per Google, it's formed by two protein monomers.
Exactly. A dimer is made up of two subunits together. Basically, a myosin is a almost golf club shape protein that has two parts. It has a long tail and a head. So basically what we have here, we'll cheat and draw it up here at the top. It has these two intertwining tails. And then it has an enlarged head region. So basically it looks something horribly like that, although a lot better. With these, hundreds, if not thousands of these are put together. And the tails all together form a big, thick, massive protein that actually is called the thick filament. So when we talk about these proteins, they're thin and thick filaments, and it is the myosin that actually produces this big, huge, thick filament. But what's special about myosin is the head. The head of the myosin is the motor. Remember, as we talked about, motors convert chemical energy into mechanical energy. And when it does that, it basically, let's do it this way. It uses the energy from an ATP to pivot. So basically what happens is this head basically pivots forward. Basically it grabs and pulls. Now notice, I'm sitting here in my chair and I'm grabbing and pulling just like the myosin heads do. So why aren't I going anywhere?
You're not grabbing onto anything.
Right. To be efficient at moving, I need to be able to grab onto something. And that is where our second contractile protein comes in. The second contractile protein is your friend and mine, actin. We've talked about actin before. Actin, it turns out, is also a dimer, but it is a globular dimer, basically made up of two globular subunits. So like that and like that. That's it. That's actin. Now, the nice thing about this is we can take these actin filaments, actin proteins together, and very poorly here, because again, my online drawing skills with this whiteboard are somewhat limited, but you get the idea. Basically, I can put a bunch of dimers together and I can make a big, long thread-like filament. So again, this is how it would look, but for simplicity's sake, I'm just going to draw it as a line. So again, we have this big, long line of actin that I can form. And these actin dimers have some handholds on it. And those handholds allow the myosin to grab and pull. Just like a ladder. A ladder has handholds on it where you can grab and you can pull, and as you grab and pull, you would go up the ladder. So this is a dimer, and it has the binding site for the myosin to grab. All right. And those are our two contractile proteins. Questions on that? All right, stunned silence means we understand. Quick, go forward and do it more quickly. Excellent. So let's talk about our regulatory proteins. Regulatory proteins are the ones that, again, turn the contraction on and off. Myosin wants to grab onto actin and pull on the actin. And as long as ATP is available, and how often is ATP available inside of a cell?
All the time.
All the time. As long as ATP is available, which is all the time, it wants to grab onto the actin and pull, causing that contraction. So we need a way to control when myosin is able to do that. And again, 2 years here I've been teaching online, feels like 40. I still have to work on my examples because normally we're sitting in the classroom over on campus. And I remind people that just down on Madison, a couple blocks from where we are, is a McDonald's. And usually at least half the students in class have gone to that McDonald's at one point or another. And if you've gone through the drive-through like I have, then one of the things you may have noticed is right next to the drive-through is a ladder that you could actually use to get onto the roof of the McDonald's. And it looks very inviting. And how many of us have actually gone up onto the roof of that McDonald's?
Probably none.
None. Why not?
Isn't it locked? Doesn't have like a.
Yeah, exactly. It isn't so much that it's locked, but
like
if
a
you,
plate
and
thing
again,
on it?
what they have is this big, huge sheet of plexiglass that basically sits over the top. And because it sits over the top, you can't grab onto the handrails. And that's exactly what these regulatory proteins are. There is a big, large thread-like protein that sits on top of the actin. And the long thread-like protein that sits over the binding sites on the actin is called tropomyosin. So there's this big, long thread-like protein called tropomyosin that basically sits over the top of the actin, blocking the binding site, not allowing the myosin to grab onto it. And great. As long as it's over there, the muscle is relaxed because it is not going to be able to contract. However, eventually we are going to want the muscle to contract. So that means we need a way to move it out of the way so that the myosin and the actin can interact. And as it turns out, there happen to be some handle-like proteins on top of this tropomyosin. And this handle-like protein can move the tropomyosin out of the way. And this handle-like protein that can move the tropomyosin out of the way is called troponin. Right. Basically, what will happen is troponin undergoes a conformational change, which again is a very fancy term. What does it mean to undergo a conformational change again?
Change shape.
Yeah, it just changes shape. And when it changes shape, it rotates the tropomyosin out of the way. That sounds like a pretty wonky thing. Guess what it is that makes troponin change its shape and move the tropomyosin out of the way?
Calcium.
Calcium, exactly. So uses calcium to change its shape, to move out of the way, and that allows the muscle to contract. All right. Questions on that?
Nope.
All right, then let's talk about our structural proteins. Structural proteins are what give the shape to the sarcomere, give the shape to the myofibril, give us our precision, give us our stripes. The first of these, what colors haven't I used? I haven't used green yet. The first of these is an important protein called the Z disc. The Z disc is a large protein that forms the boundaries of the sarcomere. Right? They're kind of like the wall. I happen to have a wall right here, and I share that wall with my daughter. Notice not only does it form a boundary for my room, but it also forms a boundary for hers as well. So when we look at this, that Z disc forms a boundary for this sarcomere, but it will also be a boundary for the next one as well. And I know I didn't make this one the same size, but you get the point. So they are the boundaries of the sarcomere. So notice here, these three Z discs make two sarcomeres. The Z disc is also an anchor point for the actin. So our actin filaments are going to be connected to the Z disc. And I'll just put a couple on here. All right. So there's our actin. Here's our Z disc. Remember, our myosin is going to be this big, thick filament that happens to sit here in the middle. And remember, this is going to have all those myosin heads that come off of it. And I'll just draw a couple of these. And since we've drawn them, let's go ahead and put our regulatory proteins in here as well. We know that thread-like tropomyosin is going to be sitting in between the actin and the myosin. And we know we need some little handle-like troponins on them. So we're starting to see the arrangement of our sarcomere here. However, notice I've only drawn one of these. All right, and notice there's really nothing holding this myosin in place right now. But lucky for us, there is another structural protein. And that second structural protein is called titin. Oops, spell it right. Titin comes from the Greek word for titan. And remind me again who the titans were? I know we talked about them. I know you weren't expecting to have Greek philosophy when you took anatomy and physiology.
Basically, they fought against the gods and lost.
Right, exactly. And they were these huge, massive beings. Remember, we talked about Atlas is a titan, and his job is to hold the world on his shoulders. They were massive. Well, this protein, when it was discovered, was the largest protein that had been found in the body. And so because of that, they gave it this name. What's interesting about the titin is that one of its jobs is to provide some stability for the myosin. So what happens with this protein is that it lines up along the base of the myosin, helping to hold it in place. But then something really interesting happens to the titin. It has this big, huge coiled structure that it has before it anchors to the Z disc. Kind of almost like a slinky shape, right? Anyone here know what a slinky is? I know many of you are millennials. I don't know if you know what a slinky is, but basically back in my days, that was entertainment. It was this big coiled wire that you put at the top of the stairs and it would walk down on its own. And if I had a slinky here, and I don't happen to have one, but if I had a slinky here and all those coiled metals, one of the things that I would be able to do was stretch it out. And when I stretched it out, would I damage it?
No.
No. And when I let go of the slinky, what would it do?
Go back to its original.
Go back to its original shape. Wasn't those two characteristics of muscle cells? Didn't we say muscle cells have extensibility?
Yeah.
Where they can be extended without damage, and then elasticity, where they go back to original shape? Yeah, well, this titin protein is what provides it. Now, just like a slinky, is it possible to stretch a slinky out to the point where it becomes damaged?
Yeah.
Yes. Is it possible to stretch a muscle out to the point where it becomes damaged? Yes. But the extensibility and elasticity, so not only does this stabilize the myosin, but this is what gives the muscle cell some extensibility and some elasticity. Allows it to be stretched without damage and allows it to recoil back to its original shape. All right. But notice, as I've mentioned, I've only drawn one of these so far. A single sarcomere is going to have dozens of these myosins, dozens of these actins, dozens of these troponins and tropomyosins, if not hundreds of them, all together inside of them. And they all have to be perfectly aligned. So to perfectly align these, I'm going to need a third structural protein. And that third structural protein is called myomesin. Myomesin basically forms three interlocking lines of protein that Don't just stabilize, oops. Oh, well. Don't just stabilize the myosin, but it stabilizes all the myosin in a sarcomere and connects it to the plasma membrane. Because remember, one of the things we saw is not only are the proteins perfectly aligned in one cell, but they're also perfectly aligned to the proteins of the cell next to it. And so we'll draw that here in simplicity as just 3 straight lines. And I'm going to try to move those so they're a little bit more towards the center. In the center of my sarcomere. And there you go. Two contractile proteins, 2 regulatory proteins, and 3 structural proteins. The 7 proteins we need to understand to make up a sarcomere. And then, of course, there's this repetitive pattern. So, of course, I have more myosin on this side and more actin, and so on and so forth. And again, if you don't like my art, let's take a look at the pretty pictures from your textbook. Three main types of proteins. First are contractile proteins. These are the ones that generate the force, produce the tension. Regulatory proteins that turn the contractions on and off. And structural proteins that maintain the organization. Here, we see our contractile proteins. Notice, as we mentioned, myosin is a dimer with 2 intertwining tails and 2 heads. They form together with hundreds, if not thousands, of other myosin dimers to form our big, huge, thick filament at the center of our sarcomere with all those heads sticking out of it. And as I mentioned, that myosin head is a motor. It converts chemical energy, in this case, the chemical energy is ATP, into mechanical energy, mechanical force of a pivot. Of course, it needs something to grab onto, and that is our actin filament. Here we see those individual dimers, subunits, and they have a binding site, a handhold, for that myosin head to grab onto. We also know that that actin is going to be connected to the Z disc. And notice also here on top of it, we have those regulatory proteins. And let's take a closer look at those. Oh, first here, I love this picture from your textbook. Electron microscopy view. When we're looking under this electron microscope, we can see very clearly the interaction between our big thick filaments, that is our myosin here, and then we have, let's do this first. The heads of those myosins coming off. So we see all those extensions of the heads, and we see they're attaching to our thin filaments, our actin. And so these are completely interlaced with each other. Myosins are surrounded, I think on average 8 actins surround a single myosin. So we have that structure and organization to it. Here we see those regulatory proteins, the long thread-like regulatory proteins sitting over the top of the actin, blocking the binding site, and that is our tropomyosin. And then we have the handle protein, that troponin, that can change its shape to move it out of the way when calcium binds to it. Lastly, our structural proteins help us to put this together to form our sarcomere. We have our Z discs, which again form the boundaries of our sarcomere. Our titin, that is that big, huge protein. And again, one of the things I want to emphasize, coiled like at the end, and then it stabilizes the myosin and actually connects to the myomysin that at the center. But, and I'll change colors just to emphasize this, it doesn't go all the way across. Both sides have their own titins that meet and connect in the middle. So it isn't one that goes the entire length. We have one protein that goes to the middle, and then the other protein goes to the middle. So two different titins there in their arrangement. They're the ones that give the extensibility and the recoil. And then on the middle, I drew it as 3 lines here. It emphasizes the interlocking of those proteins that stabilize not just all of the actin, all the myosins in the middle, but remember it can actually cut and attach to the plasma membrane, anchoring and holding it in place as well. Questions on these 7 proteins? Nope. All righty. So, as we've talked about, the sarcomere is the structural and functional unit of the myofibril. It is what allows the contraction to take place, generates the force. But notice there's also the precision of the arrangement to it. And so when we look at this, we can see how this arrangement gives us our stripes. We have this repetitive pattern of proteins that a sarcomere goes from Z disc to Z disc. That's one sarcomere. But notice that's also the beginning of a second sarcomere and a third and a fourth and a fifth. And notice that some areas have more proteins in them. Typically the area that has the myosin in it has more proteins in it. And so that is typically what we see as the dark stripe. The area that contains the myosin. Now remember, way over here is another myosin filament in this one. So from the end of this myosin to the beginning of this myosin, we have this region in here that has no myosin in it. And the area with no myosin in it is what we call the light stripe. So these are the dark stripe and the light stripe, or what are also known as the A band and the I band. Now, the terms A and I come from the German terms that were used for these when they first described them. However, if you look closely, notice the term dark has an A in it and the term light has an I in it. That is not why they got their names, but it is a great trick for remembering which is which. So the A band is the dark stripe. And again, what makes it dark is it has the thick filament. This is the part that contains myosin. Whereas the light band, the I band, the light stripe does not contain myosin. And that gives us our stripes. So far, so good?
Yep.
Now notice, there is still more precision to our proteins here. Notice, as we have talked about, our Z discs are big, large proteins that form the boundaries of our sarcomere, and that's actually something that can be seen with a light microscope. Yes, the protein is the Z disc, but the band it forms is also sometimes referred to as the Z line. And if you remember, when we looked at the microscopic view of our skeletal muscle cell, if you remember, at the very center of our light stripe was a little dark line. That little dark line you see in the middle of our eye band is actually the Z line. But it goes beyond that. Notice for much of our A band, myosin and actin overlap. But you will notice there is a very thin area in the middle where it is only myosin. And this area that is only myosin is what is known as the H band. And smack dab in the center of that, we have the M line, which is formed by the Myomesion and the myosin. I appreciate this can start to get a little confusing. But luckily, we don't have to look at these sarcomeres just in a longitudinal view. One of the things that we can do artificially, like with the drawing, would be to take a cross-section through the different regions of a myofibril and see what proteins were there and see how they're arranged. And if we did that, it can help us to understand the arrangement of these proteins. Notice, if this was the cut that I took right here, if I cut through my sarcomere here, I would see myosin and actin together, and that would tell me I was in the A band. If instead I made my cut right here, I would just see actin, and I would know that I was in my I band. If I did my cut here, I would just see myosin, and I would know I was in the H band. And if I took my cut right here, smack dab in the center, I would see my myosin and my myomesin interlocking, and I would know that I was on my M line. And that is exactly what our book has done for us. Notice here, if we were to take a section through some place in the I band, all we would see is actin. Notice if I took a section through the A band, I would see myosin and actin together. If I took a section through the H zone or H band, I would just see myosin. And if I took a section right through smack dab of the center, the M line, I would see the myosin and the actin interlocking with each other. These proteins really are this precise. Notice our illustration does a nice job of showing this. But the other thing that does a nice job of showing us is that muscle cell we were looking at. This is going to take 5 hours to load again. But as we scroll down to our first muscle fiber, notice here we see myofibrils. Here we see a Z disc and a second Z disc. So notice here, I'll draw on this one. There's a Z disc. There's a Z disc. So notice I have one sarcomere right here. But notice all my myofibrils are cut at different heights. So if conveniently enough, I had a superior view of this muscle model, muscle fiber model, I could see, for instance, an area that just has very thin proteins in it. What area, what region of the organelle do you think this is?
High band.
Excellent. And let's do this. Go ahead and erase that. This one is indeed the I band. Excellent. What about this one down here? I see thick and thin filaments. So what would that be?
A band.
That would be the A band. Excellent. So again, let's erase that and write A band. Conversely, what do we see going on here?
Only thick.
Only thick. So what does that make this?
The H band.
H band or H zone. Both terms are acceptable. And then here where they're interlocking? Excellent. So notice we can see this from the top as we're looking down from it. But also if we go back to the previous picture, we can see some hints at that. Notice we can already tell just by looking at this one, if we looked at the top of these two, notice the top of these two are going just through the thin filaments. So those must be I bands that we're looking at there. Notice this one is going through where the myosin and actin overlap. So that is our A band that we see here. Notice this one here cuts right through the myomysin. That's our M line. And notice up here, it's cutting through just our myosin. That was the H zone. So notice this shows that really nicely. Now, remember there are two cell models here, but notice with the second model, it doesn't cut it at different lengths. We can still nicely see the dark band and the light band and the dark band and the light band and how they line up with each other. They've even given us a hint of the H zone here and a hint of a Z disc there. But notice the other model does a much better job of showing the proteins, a much better job. So here we can see that sarcomere, Z disc to Z disc, the myomysin at the center, the thick myosin, and then the thin actin. So we can see this sarcomere really nicely with this. There's one other model that does a great job of showing us this, not so much in cross section, but notice we have a great sarcomere model. What I love about this model is it has a plastic cover, but notice if you look closely at the plastic cover, you can see that they've given us this yellow stripe that comes along the top, and then these dark blue, green stripes next to it with the thin lines radiating off to it. The green represents our sarcoplasmic reticulum, and this is our terminal cistern. And notice another terminal cistern. So what does this make this yellow thing in the middle?
M-line.
What is it? No.
Oh, it's the transverse tube or
transverse tubule, which collectively makes it the triad.
Right.
Right. We know those drape over the myofibrils, but if we remove that stuff, then underneath what we see is this nice model of a sarcomere. Notice we have a Z disc over here. This represents the myomycin in the center. Here are thick filaments of the myosin. Notice they've got the little heads coming off of them. Attached to the Z disc is our red actin. And notice closely on the red actin, we have yellow lines and little green knobs. What do you think the yellow lines on this represent? Tropomyosin, there you go, the regulatory proteins. And then the little green ones then must be what?
Troponin?
Yeah, the troponin, which is going to move it out of the way. You are absolutely, positively, guaranteed, 100% certain to see these models on the exam, right? And I know we're always careful when we're looking at the models because we think, well, when you look at the models, it doesn't really look like that in real life. But in this case, the model makers really have done a good job of showing us. Here's our illustration. Again, notice Z disc to Z disc is our sarcomere. The A band contains the myosin and is completely contained within one sarcomere, whereas the I band is the part that doesn't have the myosin and crosses between two sarcomeres, has the Z disc in the center. We also have the area of just myosin in the middle of our A band, that is the H band, and of course the M line in the center. And again, here we see it in an illustration, and here we actually see it using an electron microscope. Using an electron microscope, they have taken a picture of that myofibril in a muscle cell. And look how beautiful this is. We can clearly see the light stripes and the dark stripes. We can clearly see the Z discs forming the Z lines. We can even see that while there's a lot of overlap, there is an area that is a little less dense at the center. Probably didn't put that one in the best spot. That is the H zone, but smack dab in the middle of our H zone, we have that darker M line. Notice they don't do as good of a job of showing it, but here with the electron microscope, this disruption here, this is the sarcoplasmic reticulum. This is the terminal cisternae. These are the triads. This is what is separating one of these organelles, these myofibrils, from the next. And notice the next organelle has the exact same arrangement. They've got a better view of this with a colorized one. Notice here with the colorized one, we can again see the boundary between the two right here. One myofibril, the second myofibril. And notice they don't align perfectly. But is that because they're not aligned perfectly in the muscle cell? No, it's because what they had to do this was actually to be able to take a small piece of muscle tissue, freeze that muscle tissue, using a very sharp blade, slice off a tiny bit of this, put some stain and other chemicals on it, put it on in a petri dish, put it inside of this electron microscope, bombard it with electrons. And so while doing all of that, they damaged it a little bit. But even with a little bit damaged, they're still pretty darn closely lined up. And again, we can clearly see our A band and our I band, our Z line, our H zone, our M line, right? And of course, the Z discs that take us from one end of the sarcomere to the other. And of course, if we go back to our original view, again, when we're looking at just a single muscle cell, in that single muscle cell, we have a light band and a dark band, and then a light band and a dark band. And notice at the very center of our light band, we can see the Z disc. And again, notice if you look very carefully, it looks a little lighter in color, smack dab in the center of our dark stripes. That would be the H zone. Now, with a light microscope at this magnification, are we going to be able to see the M line smack dab at the center? No, but we can see everything else. And again, we see that amazingly perfect alignment from one cell to the next cell to the next cell because of proteins like the myomycin and some of the dozens of other structural proteins that give this its amazing precision. All right. Questions on that?
No.
This is it, folks. We've broken it down to the most basic bits, the proteins that make this. And now that we have the proteins, the building blocks, we have taken that wind-up clock and broken it down into all the bits. And now what we can start doing is putting those bits together to see how it works. And that is the last thing, not the last thing, but the first thing we have to do now that we've broken it down to all the bits. We have to understand how a muscle contracts. And we are going to do this all the way from the very beginning. And again, let's be precise. How a skeletal muscle contracts. Where does the skeletal muscle contraction begin?
In the brain.
Yeah. This is a voluntary action. I make the decision in my brain first. So we are going to go all the way where it begins from the decision to the point where the part of the body moves through space. This is one long continuous process. It is a long continuous process. It isn't hard. There are just many, many steps. And because there's many, many steps, we are going to break the single process into 3 steps. And are we just going to arbitrarily break it at just 3 random points? Or really 2 random points that just make through processes? No. It is important to remember this is a continuous process, but we are going to break it into 3 more easily digestible processes, 3 more easily digestible steps. Each of those steps is going to have a specific goal. And for each of these steps, we are going to learn how to initiate it. Right? How we start it. But of course, once we start the muscle contracting, do we want it contracting forever?
No.
No. So we are also going to need to talk about how we terminate the process, how we get it to end. And if we truly understand how a process works, as we've talked about many times, one of the reasons we study how the body works is so that we're able to manipulate it to our advantage. We can learn how these processes work, and then by understanding it, we can use it to improve our lives. One of the ways that we can show that understanding is by understanding what happens when we modify the process. What happens if we remove something or add some toxin or make some other change? And how will that change affect the normal process? So for each of these 3 steps, we are going to look at how to initiate it, how to terminate it, and how to modify it. And I guarantee you with 100% certainty that at least 2, if not more, of these processes will be essay questions on the exam. Guarantee it with 100% certainty. So one of the things you're going to be wanting to do over this 3-day weekend is not just to look at all of the anatomy that we've talked about, but also to start looking at some of these processes. Let's talk about these processes. Let's give names to these things. From decision to contraction is a 3-step process. The first step in this process occurs up here where the nervous system talks to the muscle cell. And step 1's name is called communication at the neuromuscular junction. Now, all of these processes have a goal. In this case, our goal is to take the language of our nervous system, neural action potentials, and use it to irritate our muscle cell, because we know our muscle cell is irritable and it is electrically excitable. And so when we stimulate the muscle cell, we produce a muscle action potential. And so that's our goal. Our goal of step 1 is going to be to convert a neural action potential, and since these are written out just below me here, into a muscle action potential. So that is the goal of step 1, and it occurs up here at the specialized structure known as the neuromuscular junction. Now, remember when this occurs, we get an electrical signal in our muscle cell, but what we want the cell to contract. So we need a way of bringing that excitation and that contraction together. And that is step 2. Excitation, contraction, coupling. We're bringing together the electrical signal and allowing it to contract. Now, why aren't the muscle cells contracting all the time?
Because we're not sending constant electrical signals.
True, but what is actually physically stopping the muscle from contracting all the time?
Takes a lot of ATP.
True, that is part of the problem. It does require a lot of fuel, and we'd be exhausted if we did it all the time. But isn't there some kind of physical barrier in the way stopping the myosin from interacting with the actin?
Tropomyosin?
Yeah, those regulatory proteins, absolutely. And that's really the key to excitation, contraction, coupling. Here, remember, we're always thinking in terms of goal. Here, our goal is to move the regulatory proteins. And what did we say we needed to move those regulatory proteins?
Energy.
Now, well, to move the regulatory proteins, remember, moving the regulatory proteins is a wonky thing. And so that wonky thing we need is calcium. We're going to use calcium to move the regulatory proteins. That moves the regulatory proteins, allowing the myosin and the actin to interact. And when the myosin and actin interact, they produce the contractile cycle, or what is also known as our sliding filament theory. Of course, our goal here is to generate the tension, move the body, with force. And this occurs, as we'll see, from myosin and actin sliding over the surface of each other, which is why it's called the sliding filament theory, but also like contractile cycle, because what is contractile cycle? What does the term cycle tell you?
And it happens repeatedly.
Process.
Yeah, it's a continuous process. Like me climbing a ladder. I grab and pull, and then I grab and pull, and I grab and pull, and I grab and pull. It's a continuous process to get up that ladder, right? And that's what happens here. We have this continuous cycle of the myosin and actin interacting, generating the tension, producing the force, right? Moving the body through space. So those are the three phases, the three steps that together make this whole entire continuous process that is a skeletal muscle contraction. Now, if it was a fourth step, what would the fourth step be?
Muscle relaxing.
Yeah,
Relaxation.
stopping the process, because once the muscle's contracting, we don't want it contracting forever. So if there was a fourth step, that would be muscle relaxation. And so we can talk about muscle relaxation as its own step, or like we also talked about, we can also just talk about how we turn on a step and how we turn off a step. So we'll think about muscle relax in both ways. We can put it continuously together as a singular process, but we could also talk about turning it on and turning it off. I strongly encourage you to take advantage of this weekend to study this material. There's a lot of material. It's very dense, but as we've also talked about, you have a lot of great resources to help you to be successful with this. Let me go to your mastering A&P. One of the things that I want to emphasize to you for this section is there is just as much anatomy in this section as there was for the skeletal system, but there's more physiology. There's a lot of material you're responsible for. And so one of the things I strongly encourage you to do is to take advantage of the resources that are available for you. Here is a massive number of them. There are the, hopefully at this point, obvious things for you. Like for instance, there are art labeling activities. So when you look at the muscular system, there's all sorts of things, superficial muscles, muscles of the abdomen wall, posterior muscles, and so on and so forth, where you can do those types of activities. Oops. Let's go back to here. However, there may be some resources you haven't taken advantage of yet. You may not have used the interactive physiology, but here under the interactive physiology, notice, lo and behold, these are directed tutorials. And look at the first three: the neuromuscular junction, excitation contraction coupling, cross bridge cycling. Those are the three processes you are going to be responsible for. And if you just want to dip your toe into this, notice there are some lab videos. And if we look under A&P Flicks, lo and behold, under the A&P Flicks, events of the neuromuscular junction. High resolution, high magnification illustration of this process.
Nerve impulses, also known as action potentials,
Can you guys hear that?
Yeah.
Yeah.
Travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plane of the skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across a synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma.
And there you go. Just that easily, it has shown us all of the steps in the process of communication at the neuromuscular junction. And from here, it's even going to show us how to terminate it, how to turn it off. These are great resources to help you to be successful with the physiology. And the physiology absolutely positively is important. However, there is also a massive amount of anatomy. And unlike in bones, where you just had to know bones and bone features, for the anatomy of our muscle cells, you are going to be responsible for many of them, for 4 things. You're going to need to be able, for all muscles, you need to know obviously their names. And you need, for all muscles, you need to know their actions. These are using the terms that we've talked about, like flex and extension, right? And abduction and rotation and supination and pronation and all those terms we've talked about. And how do you know which muscles and which names you're responsible for?
The handout.
The handout, absolutely. Notice if we go back to our modules, lo and behold, not only is there a muscle histology handout you're responsible for, but there is a muscle anatomy you are responsible for, giving us a list of the muscles and their actions. Notice some muscles just have 1 action, some have 2, some have 3, some can have 4. If a muscle has 4 actions, how many of those are you going to be responsible for?
4.
All of them, exactly. But I want you to notice something else about this list. Notice as you look at this list, some of the muscles are written in bold. And those that are written in bold also have origins and insertions written for them. These origins and insertions are the attachment points. And all the ones that are written, you need to know the origins and the insertions. Now, does that mean that these muscles down here don't have origins and insertions?
No.
No, of course
No.
not. They have them, but since they're dotted lines, since they're not bold, you don't need to know them. You are only responsible for the origins and the insertions of the bolded muscles, of the ones that are listed. A good way to learn them is to study them. You're going to be responsible for them. And conveniently enough, many of these things, like the coronoid process of the mandible, are things we've learned on the previous exam. What that means is on a picture of a model or a picture of a chart, I'm going to point an arrow at this muscle. And basically, there's 4 questions I could ask. Identify the muscle, identify the 3 actions of this muscle, identify the origin, and identify the insertion. But what this also means is on the exam, I guarantee you in the test bank is going to be a picture of a mandible. I guarantee there's going to be a picture, hold on, of a skull. All right. There's the eye, there's the nose hole, there's the ear hole, there's the big bump in the back, clearly a skull. And lo and behold, there will be an arrow pointing at that bump behind the ear. And when I pointed that bump behind the ear, I'm going to ask you what muscle inserts into that bone feature. And you're going to need to know it's the sternocleidomastoid. Like I said, the good news is these are bone features we have already talked about, but we're still going to go through them together to help you to learn them. There is a handout of bones that we are going to start going over on Tuesday. And so on Tuesday, one of the things you're going to want to make sure you bring to class, and again, you're home, so hopefully it won't be too bad, but you are going to need a red pen and you are going to need a blue pen. Because by convention, origins are drawn in red and insertions are drawn in blue. So we're going to go through the muscles you need to know origins and insertions, and we are going to draw them together. But you don't have to wait for them to start studying them. Because the other thing that hopefully you took advantage of for the skeletal system is your practice anatomy lab. Your practice anatomy lab, and since it's going to be on this exam too, if you haven't looked at it yet, the good news is there are some skulls, and that's a crappy picture, but come on, give me a side view. There we go. We can remember that again, if I have an arrow pointing towards that, that is indeed the mastoid process of our temporal bone that we can see there. But the other thing that is here, go back up to the top, go back to the home. If we look at the muscular system, yes, there are cadaver pictures that are really, really cool where we can see the muscles on the cadaver and they're useful that way. But what you're going to be tested on more is the anatomical models. So notice if you go to the muscular system and you look at the upper limb, you have pictures here that are identical to the models that we have in the classroom, which can help you to learn the muscles of the arm that you're responsible for, or the leg that you're responsible for. And there's quizzes and there's practical exams that you can take. But what's even more awesome than that is when we go to the animations, and for instance, we go to the upper limb, and we go to the bicep brachia.
Biceps brachii is the large two-headed flexor muscle located in the anterior compartment of the arm. The origin of the long head is from the supraclenoid tubercle of the scapula. The origin of the short head is from the tip of the coracoid process of the scapula. The two heads insert by a and by the bicipital aponeurosis, which blends with the deep fascia on the medial aspect of the forearm. Biceps brachii flexes the forearm at the elbow, supinates the forearm, and weakly flexes the shoulder. It is innervated by the muscular-cutaneous nerve.
What I love about these animations is they isolate the individual muscles. They show you the origins, they show you the insertions, they show you the actions. It is a great way to study this material. So I strongly encourage you, if you haven't been using these resources yet, please start now. And so that's what we're going to start doing on the next class. Lecture will be physiology, understanding how these proteins work together, talking about those 3 steps that we've been talking about, and building back up so we understand how we move the body through space. And then lab will be origins, insertions, and actions. Now, there's one more thing I'm going to strongly encourage you to focus on. On the exam, you're responsible for muscles, origins, insertions, and actions. But you're responsible for all the actions. So for instance, here with the bicep brachia, notice there were 3 actions you needed to know. It flexes the elbow, it flexes the shoulder, and it supinates the hand. Now I could brute force try to memorize that, or I could try to think of some type of common or even not so common activity that puts all of these together. And the one I always think of is bowling. Notice if I'm going to go bowling, when I'm throwing the ball, obviously I have to flex my shoulder. I have to have my hand supinated to hold the ball. And if I really want to put that curve on the ball, as I'm letting it go, I need to flex my elbow as well. So notice flexing the shoulder, flexing the elbow, supinating the hand. But I can remember that the bicep brachia allows me to throw a bowling ball. Then I can use that mnemonic of throwing a bowling ball to remember the 3 actions that the bicep brachia allows me to do. So one of the things as you're going through these is I want you to start looking at these muscles and thinking of some activities that incorporates all of their actions. I will ask for demonstrations for participation points during the lectures or during the labs as we're going through it. But you'll notice also there is a muscle activity review graded for correctness that encompasses this both ways. I give you a muscle and its actions, and you need to come up with a common activity where you would do it. And then that handout also gives you some common activities where you have to figure out what muscle would allow me to do it. So not only are these great mnemonics to help you to study the material, but I expect you to present them to me so that we can break up the boringness of going through this material, get your participation points for being active. I'm not going to bother putting you in groups for this. You don't have to be in groups for this. Just work on this on your own and be willing to demonstrate them. And it's going to help you to be successful on that graded for correctness review as well. So that's our game plan going forward. Questions on that?
So for when we're going through each of the muscles, and you mentioned, the red pen, blue pen, are we going to be using this unlabeled muscle pictures PDF to?
Yeah, so I don't think it's available yet online because I purposely didn't want you messing with it yet, but it will be it will be available when we start going through it on Monday. So if I cheat, hold on a second. Yeah, so it'll be available Monday at the beginning of class. Tell you what, I will make it available Sunday so that you can print them out ahead of time.
Maybe I'm looking at the wrong thing then, because whatever I'm looking at is already available.
Is this already available? Looks like it's not supposed to be available yet.
The origins and insertions is not available, but the unlabeled muscle pictures.
No, well, that's, yeah, no, this is what I'm talking about.
Oh, got it.
So this handout will be available beginning of class. Actually, I'll set it up for Sunday. I'll make it available. So what we're going to do is you're going to, with your red pen and a blue pen, right? I'm going to tell you that, for instance, the masseter inserts into the mandibular angle. And of course, since you know the mandibular angle is this angle of the jaw, you're going to put a little blue line right here showing that that is the mandibular angle, and that is where the masseter inserts. When I tell you that the masseter originates from the zygomatic process, or pardon me, the zygomatic arch, I should say, then you know to put a big red mark up here because that is its origin. And now we know the origin and the insertion of the masseter. And so that's what you're going to be doing. Using a red pen and a blue pen, you're going to draw the origins and the insertions. Okay?
Got it, thanks.
And I'll tell you what, since this is summer school, I'll just go ahead and release it now since we got a 3-day weekend. So if you guys want to start working on that, you are encouraged to do so. So let's just go ahead and publish that. Excellent. So bring your red pens for that. All right. One last thing. Again, hopefully at this point in time, you have figured out the things that help you to study and be successful for you. However, one of the things that I find really useful, because I'm a very visual and a very tactile learner, are flashcards. However, when making a flashcard, you have to be careful. What I find people do sometimes with these muscle lists is they'll take their flashcard, and on one flashcard, they will just put the name of the muscle. And on the back of the flashcard, they put the origin, they put the insertion, and they put the actions. And that is not a good way to study these because you need to, because I'm going to be showing you, my guess is about 25 to 30% of this lab exam will be bone pictures, where I will be showing you a bone feature that is either an origin or an insertion. And based on that, you have to figure out what the muscle is. Now, one of these days, I will copyright this and I will become rich and famous and I will never have to teach anymore. But until then, take my advice, take your flashcard and make them 4-sided flashcards. Take those flashcards and fold them in half. When you take a flashcard and fold it in half, I have a piece of paper here. I don't know what this is. Hopefully it's not important. Take it and fold it in half. What you can do on one of these, you can then write the origin. On one of these, you can write the insertion. On one of these, you can write the actions. And on the fourth, you write the name, or you have a picture of the muscle. So yes, the first time you look at it, you look at the name or you look at the picture and you think of what it is. And once you say, okay, that is the masseter, then I can think of the origin and I can think of the insertion or I could think of the action. So the next time you go through, you look at the origin and you think, all right, what muscle originates from the zygomatic arch? Oh yeah, that's the masseter, right? And you can think of it that way. Or then the next time you look at the insertion, what muscle inserts into the mandibular angle? And you go, yeah, that's the masseter. Or you look at what muscle elevates the mandible. And then I think, all right, what's one's responsible? Oh, that's the masseter. So you want to be able to look at any one of these 4 pieces of information and come up with the other ones. So having a 4-sided flashcard is a great way to do that. Of course, if you don't like making your own flashcards, I will remind you also that in your Oh, hold on. Oops, wrong one. In your MyLab and Mastering. In your study area. Practice Anatomy Lab flashcards. Using the anatomical models, you can actually identify bones, muscles, muscle system, upper limb. I can actually pick the muscles on my list that I'm responsible for. And then I can actually review these where they'll give me the quiz, having to identify the muscles that I'm responsible for that lift, and so on and so forth. It is a great tool. These can actually be downloaded that you can take that way. There's self quizzes. There's an actual quiz. It is a great way to customize this just for the material that you are looking for. You paid good money for these resources. Make sure you're taking advantage of them, because the point is to help you to be successful. So again, notice right here from the start of the PAL, mobile flashcards allow you to customize it, download it to your smartphone or tablet. Great resources here that if you haven't been taking advantage of, please start taking advantage of it now so that you can be successful on the exam, because we're running out of points. We're halfway through the class now. So you want to maximize your points, and so you want to make your studying as efficient as possible. All right. Excellent. Hopefully that was enough to scare you guys to spend this nice 3-day weekend. Yes, with a little downtime, but again, take a good 15, 20-minute break, and then I want you spending the rest of the 3 days studying this material. All right. Have a safe and fun 4th of July. Mostly fun, but also mostly safe. And I will see you guys on Tuesday, the 6th. Have a good weekend.
Thank you.
All right.
You
Bye,
too.
guys.